So we have 1 more minute to relax until we begin. For everybody that's joining from around the world, welcome. We're so thrilled to have you all. Incredibly excited to have you all here. And all right, why don't we get started? Okay. So for everybody in the room, welcome to the 2025 edition of How to Grow Almost Anything. So delighted to have you all here, both in person from MIT and Harvard, and also for all of you that are joining from around the world. It is an honor to have you all here with us. So we have a very, very packed agenda for today. So let me just start by sharing some slides of what we are going to do. All right. So let me head over here. For a global audience, if I hit play, tell me if you still see these slides, hopefully. Give me 1 second here. Are you all seeing slides?
Yes.
Great. That is, in fact, the Jurassic Park theme song in the background. But welcome, everybody. And also, again, as I mentioned, welcome to everyone who's joining from around the world right now. Just a quick bit of background for you all here. I think one of the special things about this class, we have it as an MIT Harvard course, but we also open it up to students all around the world. And over the past number of years, we've had hundreds of students participate in the class. Globally, this year we've got more than 1,200 of you that applied to be part of the course, which is pretty amazing. And we'll see how many of you are arriving today. I guess around 370 right now, but we'll see how things progress over the class. But again, so honored to have you all here joining us. For folks that are joining online, please stay muted, keep your video on if possible, and then also use the naming convention shown. Ideally using the first name and last name from your application. And again, TAs, please share that as well. And so for those of you that may be new to the Media Lab, how many of you are new to the Media Lab? A number of you. Okay, so the Media Lab, and for those of you that are joining from around the world, we do a tremendous amount of research here, focused on science and engineering and technology, but also in art and design. And I think one of the really special parts historically about the lab, the Media Lab has really, I think, pioneered the development of looking at different types of technologies, being it coding and computation or digital fabrication, and then developing learning experiences around those and then scaling them globally. So many of you may be familiar with Scratch, developed by our colleague here at the Media Lab, Professor Mitch Resnick, which again is very recently has had over 100 million users, which is just an outstanding, unbelievable number. And globally, there are these wonderful computer clubhouses where folks can engage in Scratch and engage in learning. Similarly, the course How to Make Almost Anything. I'm curious how many of you have taken How to Make Almost Anything? A few of you have. Okay, wonderful. As a really remarkable digital fabrication course, and has also been scaled globally through the Fab Lab network. And through How to Grow Almost Anything, we have been developing a similar type of synthetic biology course and also scaling it through a network of both community biology labs and also other types of wet labs that are located around the world. So today, what we're going to do is we're over the next approximately hour and 20 minutes, we're going to do a very deep dive into what this course is all about, what you all will be getting yourselves into. We're going to have some quick introductions from the faculty instructors, and then all of you actually are going to have an opportunity to very quickly introduce yourselves. And anyway, so we'll go into the deep dives of what the course is all about. We'll take a 10-minute break, and then in the second half of the course, we actually have a few student alumni who are going to come and share their experiences, and they'll be available to answer some questions as well. I'll be giving a lecture in addition on principles and practices. So fundamental ethics around synthetic biology, why we should grow almost anything. And then we'll close out with any remaining time with some Q&A by myself and George and Joe and any of the TAs that are present. So one of the really special things about the class is the really remarkable set of faculty. So again, in addition to myself, Professor Jacobson and Professor Church, there's a really kind of sparkling set of faculty from all around the world. For those of you that are in the field of synthetic biology, may recognize some of these names and faces. Truly pioneers in their fields, all of whom will be sharing their latest research as a part of the cutting edge of synthetic biology. And so that's our faculty cohort. And what I'll do right now actually is I'm going to stop sharing these slides, and I'm actually going to turn it over to George and to Joe just to introduce themselves and give a quick welcome. So George, I'm going to spotlight you first, if that's okay. So you know that you are on the big screen now in the classroom here. So George, it's so great to see you. And yeah, we would love to have you just share a few opening and welcome remarks, George.
Yeah, so I'm George Church, professor at Harvard and MIT, and quite engaged in the media lab and that community above and beyond this course. This is an absolutely terrific course. I think in part, lab courses in general tend to really bring you into the subject matter, even if you mainly interested in it theoretically. Getting a little bit of lab experience is great. And I think that having this worldwide community with a fair number of shared robots and robot protocols is important. I think our emphasis on ethics and safety, not just as a consideration, but as a opportunity to engineer, is a nice piece of this course. The interdisciplinary nature of the student body and geographical diversity are all very attractive, I think, to you and to me. And the final project, I think, it really stands out to me as something. It's we didn't have this from the beginning, but I really like it. I think it's a great opportunity to have a community goal that is more ambitious than you would do if you had to do it by yourself. Yeah, those are some of my opening things to say, but I welcome you all, and I look forward to interacting with as many of you as possible.
Okay, George, big round of applause for George. George, thank you so much. So you can bring your chair over. George, hopefully we'll get you in Adams at MIT for one of these classes as well, but it's so great to see you. So I'm now going to turn it over to my colleague, Joe Jacobson. And Joe's got a couple of slides just to share as well as part of the opening. And just give me a moment here as I make sure I share the right slides. Sorry, give me a second here. Okay. All right, and everybody globally, if I just make sure if I press play, that this also works. Give the computer a second to think. Yes. Okay. Are you all seeing slides?
Wrong presenter view.
Okay. Yeah, let's hop to see slides, Joe. So I think what we'll do very quickly is if you can just present from the this view here. Okay. All right. Yeah.
Fantastic. Exactly. Go ahead. Great. Okay. Good afternoon, everybody. My name is Joe Jacobson. I'm a professor at MIT. It's always one of the best days of the year, I think, the first day of how to grow almost anything. Part of the reason it's one of the best days of the year, I think, is just the excitement and opportunity of what's possible in the synthetic biology space. And I just want to give a little bit of background into why many of us that didn't start in synthetic biology, I suppose, like many of you, got really excited about this field. And so I want to start kind of by analogy with silicon. I don't know if there are any chip designers in the room. Anybody here designed a chip before? No. But as you all know, silicon has been kind of front and foremost, certainly in the last couple of years. Many of the things that we do, obviously powered by silicon. And one of the reasons that silicon is so powerful is that it harnesses the ideas and concepts from a very large number of people. We have in silicon something called an IP core library. It's a library of different parts, millions of different, they're called IP because they're intellectual property, they're IP cores, things, concepts that people have come up with, things like adders or MCUs or different parts of GPUs, for instance. And to build a chip, the way that's done is you suck down cores from this IP core library, from the intellectual contributions of thousands of people around the globe, and you can assemble those, you can compose those into a chip, and they are pre-composed to work together, to function. The outputs of one can feed the inputs of the next. They're what are called composable. And with that, we can build very sophisticated things like GPUs, CPUs, and we can then go to a fab. These are things like TSMC or Intel, and make these chips in a very short order period of time. A couple of weeks, we can make a chip, cut it to working silicon, and we have our product. And the number of possibilities here scales as the number of cores that we have, which, as I said, are millions, raised to the number of cores that we have on a chip. It's a very large set of possibilities. And unbelievably or amazingly, it's not the only system of its kind in the world. And the other version of this system is what biology has given us. This is synthetic biology. This is, instead of circuits, electronic circuits, we have metabolic circuits. These are systems of genes that are composable. I can take a gene from a given organism that might be a sensor for a certain molecule in solution, and I can couple that to a gene from a totally different organism, which might be an output, something like a fluorescent protein or an enzyme that makes a new chemical. And the set of possibilities here is equally large. It's nearly infinite. And I think this is why many of the people that have gotten into synthetic biology, not just native biologists, but people that are interested, as David said, in design, in art. It's that level of creativity is not very common in the things and the systems that we deal with, and the amount of possibility and the amount of power that's possible in synthetic biology, I think, has been attractive to many people. So I just want to give a little bit of a taste of an example of kind of how sophisticated this is, and how much of an analogy is present between the power of what we can do in the computer space and what we can do in synthetic biology. So this is a circuit called a ring oscillator. It's a couple of transistors that are connected to each other. The output of one transistor feeds the input of the next, and the output of that then feeds the input of the next, and the last one is tied back to the first. And this is the very first circuit. When you're building a billion-dollar chip fab, the very first circuit you build is this ring oscillator. Because what this does is it creates a sine wave here. And if you take the Fourier transform of that, you can look at the uniformity, how good each of your transistors is. And amazingly, we can build the same kind of logic out of biological parts. This was a kind of groundbreaking paper from Mike Elowitz, who at the time was at Princeton. And what he did is he made a plasmid, which was a set of genes, and a gene is a piece of DNA that codes for a protein. And we might think of proteins as the structural material, the thing that we build things out of. But many of the genes that are in biology code for proteins whose purpose is not to be structural, but is to be logical. And so in this circuit, this gene codes for a protein, and that protein doesn't go build something. It then turns on or off the next gene. And that gene in turn codes for a protein, which turns on the next gene, and so forth. And it's really an exact analogy to what we build in electronics. And then finally, one of these genes we're going to tie to something that lights up, a green fluorescent protein. And amazingly, when you build this circuit, you can see here's the output of this circuit. It's a sine wave. It looks like this sine wave. The time scale is different. It's a much slower time scale. And there's one other thing that's different about this sine wave than this. What's different? What's different about this?
It's growing.
Growing, yes, thank you. It's growing. So this is kind of this is kind of the amazing thing, is that you can code this program into a cell, and the feature that we yet have not replicated in engineering is self-replication. David, does this work here?
It should, yeah. Let's see. What's did it earlier?
Okay. In presentation mode?
Yeah, in this mode. Well,
Okay.
this is always how it goes.
Okay, all right, fine.
Maybe
I'll just check.
X out the security warning references. to-
Oh, thank you. That's exactly what it is. Oh, great eye. Enable content.
Nice,
That
thank
is
you.
it, Patrick. There it is.
Fantastic,
Perfect.
thank you so
Patrick,
much.
thank you so much. Great.
So here's an amazing thing. You load this program to a single cell, and while the program is running, the cells are also replicating. Now, one thing that you can notice here is that you've got a whole bunch of cells running this program. They start phased up or in the same phase, but eventually, very quickly, they become out of phase, meaning when one of these cells is on, another one is not on. And so you can add a little bit of to that circuitry to sync up these cells, what's called quorum sensing. You can have these cells also make a molecule that is shared between the different cells. And so after a few cycles, they all start to sync up. This is kind of an amazing kind of sophistication just in a couple of genes that we can build. So these are metabolic circuits. And we can start to think about things that become very sophisticated. So instead of just a single circuit, we can imagine things like what we have in electronics, a programmable gate array. A gate array is when you're building your iPhone, before you have all of your custom silicon, you come with a universal piece of logic called a gate array. It's got lots of different gates on it. You can program it to carry out a function that is particular to that new product. And we can imagine things like that in biology. We can imagine an entire organism that's got many, many genes in it that are not on, but they are programmable, meaning we could program them to come on. So it's like a library of possibilities, a library of genes. And you could imagine programming this with systems like CRISPR, for instance. So you have a set of guide RNAs that you load into the cell, and those guide RNAs would tell which program you want to run in this kind of universal gate array. There's two things that I just want to highlight that are coming down the pike in this course, and these are really new additions over the last couple of years. The history of synthetic biology has really been what I've just shown you. It's been taking genes from all over biology, existing natural genes, and recomposing them in ways that have never been composed before to build new things. But very recently, in the last couple of years, we've been able to go beyond that in two different directions. So one of those directions is at the molecular scale. In the past, we've been beholden by what nature, the parts that nature has given us. Very recently, in the last couple of years, and the Nobel Prize was won for this in this past Nobel Prize cycle, we've been able to design functional proteins from scratch for the first time. And this is a kind of the inverse problem of the protein folding problem. If you think about the protein folding problem, you go sequence a genome, and that codes for a protein, and then you ask what three-dimensional shape does that fold up into? The inverse problem is the design problem. I have a shape that I want to make, a certain protein, and the shape of a protein implies what its function is, either an enzyme or a binder to another protein. So I start with a shape, and now I ask the computer what sequence of amino acids will, when I string them in a single line, will now fold up into that functional protein. And very recently, we've been become adept more and more at being able to design proteins, and that's now part of this course, and I think a really exciting kind of future of the course. And then the last part of that of this expansion and being able to go beyond what's been possible before is, instead of making things that are just a single cell, starting to make multicellular things. And if you think about it, nature's really remarkable. We all start from a single cell. That cell then divides into two, into four, but eventually we start to differentiate hands and eyes and features and all of those. And the question is, how does that happen? What's the program? How do I write a program that goes from a single cell, all running the same program, but nonetheless, they start to differentiate. This is an area of programming called cellular automata. And we've known how to go from the rule to what the final shape is. We haven't known how to go from the shape to what the rule is. And so we haven't really been very good at programming this. But recently, again, and this is really a kind of a breakthrough algorithm from one of the groups at Google. We've been able to do a process in cellular automata where we can start with a target shape. Let's say you want to make a letter on the wing of a butterfly, for instance. So what's the program that you would write in order to make that happen in biology? Remember, all the cells have to run the same program, but nonetheless, they have to differentiate. So you start with a single cell, you divide, but let's say you're running a little circuit in there that's measuring the number of nearest neighbors. Eventually, you've got internal cells and external cells, and that's one symmetry breaking. You then can throw on some other symmetry breakings. You can set up a system of coordinates and axes, and then you can start to make patterns and differentiate and make multicellular things. And these two directions, going down in scale, molecular scale, and going up in scale, multicellular, are things that we're just able now to do, and really excited to be able to offer some of this in this course that you're about to take. So quick intro. Thank you, Dave.
Perfect, Joe. Thank you so much. And that was a great, I think, overview of some of the metaphors and concepts that we'll touch on in the class. I'm going to keep moving along now, and actually we're going to start doing some introductions, including from yourselves. So what we're going to do now is this class, as you will all see, is not possible without like an army of TAs that are working behind the scenes to make all of this happen. So we've got a really amazing crew of around 16 TAs that are here at MIT and Harvard, many of whom are in the room, and I'll have you all introduce yourselves in just a moment. And then we also have almost total 80 TAs that are also supporting the virtual students that are from all around the world. So, it's a really tremendous crew. I'm going to say a little bit more later about how this is all structured globally for our listeners. But for our in-person crew here, if you're a TA, can you just raise your hand? So we got a few of you. So if you all don't mind, if you all can introduce yourselves, you can just say your name and your department. Very quick history of How to Grow, like if you're a student or a TA. And then also extremely fast, a fun fact about yourself. And all of you are going to have to do this, and it's got to be really fast. So very, very thinkable, and you're all going to have to do one. So Subin, why don't you go ahead and start?
Hey guys, I'm Subin.
Hey, David, can you turn on your video so we can see remotely? Because we can't see anything, any humans.
All right, cool. Can you see the humans now? Oh, not yet. What happened to the video here? I think we lost the owl. Hold on. Let me
It's okay. You guys have more time to think of your fun facts. So.
Okay. All right. Go ahead, Subin.
Great. Hi everyone. I'm a senior at Harvard studying bioengineering. I actually joined How to Grow my freshman year. So man, this was 3 years ago. Wow. Like time has flown. Really enjoyed synthetic biology. I originally was studying mechanical engineering and switched over to bioengineering. So found this really fascinating field. So been working on these kinds of projects ever since. Fun fact, I fly planes in free time. So
amazing. Thank you. That was perfect. Patrick, why don't you go? Actually, Ice, sorry, go ahead.
Hi everyone, my name is Ice. I'm a postdoc here at MIT. I was a student last year, a cumulative listener student from last year, and I'm interested in synthetic biology in general. Apart from want to be a synthetic biologist, I want to be a comedian.
All right. Thank you, Ice. Patrick.
Hi, I'm Patrick. I'm actually a professor at Yonsei University, but I started off as a postdoc with Dr. Church and was a TA for this class last year. And now I'm starting a node for How to Grow Almost Anything in Korea. So I'm here temporarily until I go back to Korea and start this thing, and also my lab. You'll hear more about the global nodes later. So
yeah.
So I'm not sure about fun facts, but
fast, anything.
I have 2 non-working mode tests I'm working on.
There you go. Thank you. Perfect. Ren.
Hi, I'm a PhD student in biological engineering, actually co-advised under David. I am the second year in my PhD, and my fun fact is I am currently learning how to blind solve a Rubik's Cube.
Wow. All right. Very nice. Next TA. Yes, go ahead, Anna-Therese.
college last year, so throughout my college years, I was an RA in George Sherford's lab. Currently I'm an RA at sness, and I was a TA last year in a class, continuing to be the TA this year. My fun fact is that I was in the site, so I figured out these people about my today.
Wow. All right. We will call you on that. Not right this second, but later. Kana.
So I'm mentoring on Spain. I love this country. I'm right now. And my background is in art and design and so actually I'm a brand new student to biology and yeah, I think that I do yeah, my background is how can you name the video and the
Okay, welcome. Itamar. All right, fantastic. So keep the peanuts away from Itamar. Who else in that? Who else is a TA in the back? Who in this?
Ronan.
Oh, Ronan. Yeah, go ahead, Ronan. Wow. Okay. Great fact. Thank you. Lenny.
Hi, everyone. I'm currently a second year master's student here at the Media Lab in the SFL group. I think about infectious diseases, also protecting them earlier, preventing them in the first place. I did how to grow last year, decided to return this year as a TA. And the fun fact is that after like over a decade, I have swim lessons again.
Ooh, did you pass the MIT swim test? All right. Jiming.
Hi everyone, my name is Jiming. I'm a third year master's student in bioinformatics from Johns Hopkins, and I'm also a visiting student at the George Lab. Doing biology research. Fun fact about myself is I actually majored in humanities and
Oh, wow. All right, very cool. Any other TAs in the back? I think are there any other TAs that did not introduce themselves? Okay, so TAs globally, please introduce yourselves in the chat for folks there. And so now we're going to go rapid fire around the room for all the students. Okay, so just say your name, your department, your year, and then a very fast fun fact about yourself. And we're gonna optimize for speed. Okay, so we're gonna go quickly. Are you ready?
Yeah.
All right, let's go. Over here.
I'm Shrika. I'm a junior at MIT studying computer science. My fun fact is that I can speak 6 languages.
Wow. Okay. So you and then I think under the Pi recitation, we can do some language conversion there. Yeah, go ahead.
Spruce Physics Junior Conjugal Penguin.
Okay, amazing. Great. I love it.
My name is Nick. I'm actually doing a gap year, but next year I'll be attending Harvard. And this year I'm actually doing cancer research at the Harvard Cancer Center.
Okay, amazing. Thank you.
I had colleagues and all these lockdown experiences because I was at the lockdown.
Oh no, continuous lockdown. That's the worst. Glad you made it through in the back. Yeah. Oh, you're in Joe's lab. Okay, great.
Yeah, I'm Ricky. I'm a MSMEA legal student at Harvard Business School and the Engineering School. And I actually spent 3 years in undergrad in computer science, but I never did synthetic biology. I worked on the open trans robots, and I was like out in field studying Harvard.
Oh wow! All right, that's so cool. Well, Drew is a very old friend, colleague, part of our How to Grow class. So that's awesome. Yes, next.
I'm Dave. I'm software physics. And fun fact is that I work at ICT.
Oh my gosh, we are glad that you're recovered. Next, yeah. Wonderful. Thank you.
Is there any chance any of the folks that are in the room could actually speak loud enough for the folks that are virtual to hear?
We're sorry about that. We don't have a good microphone. Our owl crapped out on us. So just if you can just be patient in the class.
Another suggestion, David, is that people with laptops could use their laptops because those have microphones. They could share them with people next to them. Just a thought.
Yeah. Thank you, George. Yeah, unfortunately, we kind of kludged something together with another laptop, so I apologize for the bad audio.
I mean, they could just log into the Zoom window.
Zoom, yeah. If you're able to, you can do that actually. But let's keep this moving. Who is next? Yes, go ahead. You want to use your laptop, bro? Yeah, that works. Oh, what? Okay, all right. Okay, grow mushrooms and slime molds at home. I will try to translate for the folks in the audience. It's perfect. Okay, great. Thank you. Is sleeping? Okay, Esther, favorite activity is sleeping. We love that. Okay, next.
Hi, my name is Vincent. I study public policy at Kennedy School over in Harvard.
Oh, wonderful.
And my fun fact is I went swimming in the Charles twice, both times involuntarily.
Involuntarily, okay. Vincent from the Kennedy School went swimming in the Charles River twice involuntarily. There's a story behind that we'll learn about later. Yeah.
Schools I covered, my confidence is that I speak 3 languages.
Oh, 3 languages, 6 languages, can recite pi perfectly. This is great. Okay, up next here. Okay, Mariam from the smallest island in the Middle East. Thank you. Computational Agnes doing computational knitting. Thank you.
Hi, my name is Manuel. I'm a freshman in Harvard. I'm studying bioengineering. And my fun fact is that my home state, I'm from Mexico, right? Has the same name as the hot sauce called Tabasco.
Okay, Manuel's from Tabasco. Thank you so much. In the back, and why don't we start here? Yeah, good. Okay, we have a student with a dog who can meow. Okay, so trans species things happening. Yeah. Cyrus.
Hey, I'm Cyrus. I'm a researcher at the media group in Vienna. And my fun fact is that I guess I'm the guy who's always takes for plants.
Yes. We love, and Cyrus also has been a lecturer actually in the class before joining the lab as a student. Cyrus, great to see you. Yeah, go ahead.
Hi, Peter.
Okay, wonderful. Can you guys type into the chat what they're saying? That'll be a great way for the listeners to follow along. That'd be awesome. Thank you. Great. Okay, James, Boyden Lab, ambidextrous chopstick holder. Fantastic. Yes.
Hello, I'm from I run on building a series of machine in Lab, and I built 2 mobile lab for the twins, and fun fact, I'm a twin.
You found that out you're a twin?
Yes.
Okay, cool. Builds Fab Labs and is a twin. Okay, wonderful.
Is that I am part of Biocluster here, which is actually one of the global ones for how to grow, and I've taken half of the class.
Great, Kat. And we see Georg in Tokyo right now. Very excited that you're here. Awesome. Yes, in the back.
Hi, everyone. I'm Georg. I graduated from the Kennedy School last year in tech policy. I'm a researcher in venture capital at Harvard this year. And fun fact, so I know 4 or 5 kinds of swordsmanship in Japanese to European.
You say swordsmanship?
Swordsmanship.
Wow, different types of swords. Okay, that's amazing. We've got like 9 languages, 5 swords, and pi. All right, and then yes. Yes.
first year at MIT, we can find out there being computers and AI. Sometimes we do that. Like playing battery games, like Go or like first time, but the only kind of always play is chess.
Okay, we love it. Thank you.
Alex, freshman season, yes. My fun fact is that I really enjoy cooking Chinese food.
Okay. All right. Wonderful. The problem is when you say things like that, you're gonna have to cook for the entire class. So it's not that, it's just forewarning. Yeah, in the back. Yes.
About 3D printing of seeds.
3D of seeds? Oh, plants. Oh, okay. Very cool. Thank you. Who else in the back? Yeah, go ahead.
That is, my name is the first 3 letters of fantastic.
Fan is the first 3 letters of fantastic. That's great. Okay. Anybody else in the back did not have a chance to go? Yeah, go ahead. $250,000 minutes of music. Top genre? Indie. Okay, great. Did everybody go? Did we get everybody? Oh, in the back. Yeah. Okay.
My name is Alex. I'm a senior in the at MIT, and my fun fact is I've met a bunch of prime ministers, Scandinavian countries.
A bunch of prime ministers doing what? Scandinavian countries, specifically Scandinavian countries. All right, you're gonna have to share with us, not now, but why? That's got some good backstory there. I'm Joffrey.
Yes, hi. I'm Joffrey, and the 3rd year undergrad in biological engineering. And my fun fact is I have a collection of vintage MIT books.
Ooh, how vintage?
I think oldest one is probably from like 19.
Oh, that's pretty vintage. Okay, that's good. I thought you were gonna say like 2000 or something, and that would be very disappointing. And anybody else? Yeah, go ahead. Last 2.
My fun fact is, I rode the same road as your 28 consecutive times.
Wait, which roller coaster? Okay. Okay, amazing. And then last one, yeah. Is what?
Pokemon Go.
Pokemon Go. Are there any here right now? I was just curious if you're keeping track, but okay. Did everybody have a chance to go? All right, give a big round of applause for yourself. And that was great. I really, really appreciate that. So again, a really, really big diversity of backgrounds in the room. And again, thank you all for sharing a little bit about yourselves. So we're going to just finish up with a bit more in terms of the content of the class now that we have introduced ourselves. And for the students that are from around the world, I see you are already introducing yourselves, which is great. So please continue to do that. Again, this is as we've been doing here, share your name and department, where in the world you are, and then a fun fact about yourself. So please do that. Also, I'm curious if colleagues from Thermo Fisher are on the line right now. Dale and Andrew, if you guys are there, feel free to unmute yourselves if you're there. There's a lot of people in this classroom right now, so it may be a little hard for me to find you. But Andrew and Dale, if you are there, feel free to unmute yourselves and speak. TAs, is there an Andrew Yu or a Dale? Okay, all right. Well, we will thank them anyways, but this class would not be possible without the very, very generous support of a number of really amazing companies that have been sponsoring us and working with us over the years. Twist Biosciences, Emily LaProuse will be here next week, but has been providing DNA synthesis for the course, really incredible. OpenTrons has been providing us robots that we've been able to distribute around the world. Thermo Fisher has been helping us with materials and reagents and equipment, and Nuclera also with digital microfluidics. So just really, really grateful to all of those companies for their support. And just very briefly, just as a little bit more context, Joe, I think gave some great analogies between computation and biology. But just to give a little bit more background around, what this field of synthetic biology is. Just to say historically, humans, we've been editing and working with the natural world for, tens of thousands of years, from domesticated animals to crops and agriculture. I think the ability to really manipulate and work at the molecular scale was really transformative, and our ability to read, write, edit DNA has been a really critical set of technologies that have been developed over many decades. DNA synthesis is one that we're going to focus a lot on in this class. And part of what's kind of amazing about DNA synthesis is, in contrast to the earlier history of genetic engineering, where researchers had to work with samples from nature, you'd have to go collect DNA from a strawberry or for humans or, whatever kind of sample in nature. With DNA synthesis, we truly have the capacity to design something in a computer, have a machine that can actually print out de novo brand new molecules of DNA that have never before existed. So a really transformative technology. And, one thing about this particular class, this is a figure showing DNA across scales. We're going to look at DNA across all of these scales, from individual genes, which may be hundreds or thousands of bases. Joe's already talked a little bit about the ring oscillators and this idea of genetic circuits. We're going to spend a lot of time on circuits. And then we're also going to get into the space of full genome rewrite. So simple single-celled organisms and base around megabases or tens of megabases. So this full spectrum of DNA synthesis we'll talk about, and you will all have the chance to work with actually in this class. So I'll point you all to a historical document. Again, one of our colleagues here is from the ND lab. Drew ND, who was formerly a professor here at MIT back in the 2000s, with many other colleagues around the country and around the world, we're trying to figure out what this new field of synthetic biology might be. There was already something called genetic engineering, and the early set of definitions arose from a set of workshops that were held. And the early sets of colleagues said and framed synthetic biology as having three aspects. One is, and again, all of this is about being able to apply engineering metaphors to biology and living systems. And so one part is, can we actually work with standardized components? I'm curious if any of you have been or participated or worked with iGEM, the International Genetically Engineered Machines Competition. Only a couple of you. Okay, that's cool. So one of the ideas of being able to work with standardized parts in biology was a really, really foundational idea in early synthetic biology. Another big concept is component abstraction, being able to work at multiple abstraction hierarchies, which we do all the time in computer science and electrical engineering, and then being able to decouple design from fabrication, right? So you don't have to build a computer to know how to use it. And similarly, the early synthetic biologists thought that you could really spend a lot of time just on design without actually having to fabricate. Turns out you still have to do a lot of fabrication, but these days, increasingly with automation and other tools, we are now increasingly able to decouple design from fabrication. So these are some of the early concepts. This is a really great paper. It's linked on the website, but also you can have a search for it. It's a really great read. Another set of concepts we're going to spend a lot of time on is related to the central dogma of molecular biology, which again, many of you, hopefully, if you are new to biology, you've had a chance to either participate in the bio bootcamp and or look at the materials. But this idea of DNA being transcribed to RNA and then translated to proteins, this is something that we're going to come back to over and over again in the context of the course with greater and greater levels of sophistication. Joe's already talked about the ring oscillator, but we are going to spend a lot of time looking at increasingly more sophisticated what are called genetic circuits, which again have different types of metaphorical connections to electronics. So for example, digital Boolean logic that you can actually represent in a cell using DNA. And so we'll spend a bunch of time talking about that later in the course. And as we learn how to design and build the DNA, what can you do? One of the big areas is around biological production and being able to manufacture different types of molecules. And so we have a whole class on bioproduction, and we'll also have a lab on it. But that's one of the big kind of areas is thinking about, and this is kind of one of the industrial uses of synthetic biology, which is how can you engineer organisms to produce useful types of molecules and materials. Ginkgo Bioworks is a really great story connected to students that were like you all, once graduate students here at MIT, and have since gone on to start a lot of really significant institutions in synthetic biology. But all of this kind of larger genetic circuit assembly, genome editing and building can be applied in a lot of different ways. There's big applications in the therapeutic space, being able to use these types of genetic circuits to, for example, ID different cell types and then can, for example, if they're cancer cells, kill those cells as one therapeutic modality. There's a whole giant field now of what are called cell-based therapeutics. So being able to have engineered cells that can, for example, be in the body and part of your larger microbiome that can sense and respond to different types of environmental stimuli, including inflammatory responses. So this is a really big area of medicine that's been evolving. Some of you are already likely very familiar with cellular agriculture. So being able to use lab techniques to actually culture and grow animal products without using animals. We'll have some content connected to that. And this also bleeds into the design world, being able to use things like bacterial cellulose, also spider silk. These are all different types of materials that can now be grown in laboratories and are now finding use in the world of consumer products, which again, I think some of you may have already interacted with. Of course, there's a big application as well with other types of nucleic acids, mRNA. All of us have had interactions with, at some point now, mRNA vaccines. This is another kind of connected technology to DNA synthesis. CRISPR-Cas9, which some of you many of you probably have heard of, another genome editing technology. We're going to have a lab that's focused on CRISPR, and this connects into the larger realm of DNA read, write, and edit. And as Joe has already mentioned and has been queued up, we're now in a really remarkable time where there's a huge emphasis on protein design and leveraging artificial intelligence, machine learning tools. Just in a fast few years, we went from having to have challenges predicting certain protein structures to then predicting the entire known protein universe, and now being able to design proteins on demand. And so Itamar, who was here earlier, and a bunch of other colleagues who have been working on some really nice protein design related projects and problems for you all that will be a big part of that part of the class. So, and I'll close this overview by just saying, some of these modalities and how we think about synthetic biology, there's a lot of different types of relationships with the natural world. And so I encourage you as we navigate these different tools and technologies also to be thinking about, and this will tie into the lecture later on in principles and practices, other ways for us to relate to nature. So now, in terms of the actual structure of the course, each week, what is going to happen is we're going to have a lecture in this very room. The first hour of class is going to be a homework review. And then the second 2 hours of class are going to be introducing our new concepts for the week. And we'll have speakers, again, that are real global experts in their field, sharing their wisdom with us. And then during the week, you'll be working on skills development in the wet lab. And Suvin, plus the MIT Harvard TAs in person, will be helping you all here. And globally, again, we'll talk a little bit more about the global classroom, but that will be part of the skills development that happens after the lecture. And so for those of you that are brand new to biology, and I'm curious how many of you have never been in a wet lab before? And don't be afraid to raise your hands. Okay, good number of you. So we always have a good number of students that actually have never held a pipetter before, never done anything in a wet lab, which is totally welcome. And we have a bio boot camp. We actually did it for the first time this year. We did a whole week. It was 3 different modules, so around 9 hours of content, plus many hours of office hours. So all of that content was linked in the email that I sent out. So if you're new to biology, please do review those lectures and those materials online. And then going week by week now, today is our first class, which our focus is on principles and practices, both the overview of the course, but also some of the ethics around how and why we can design and work with synthetic biology. Next week, we're going to go into a really technical dive into reading, writing, and editing DNA. So really grateful to have both Joe and George, and also Emily LaProuse, who again is the founder of and CEO of Twist Biosciences. This is Charles Darwin, and this is George. So they're like the chiral versions of each other. So really grateful again to have those three. The following week, there's no lecture. It's the President's Day holiday. So there's no formal lecture, but we still are going to have recitation and some lab work related to lab automation and cloud biology. So this will be some of your first dives into liquid handling robots and programmable automation. The next 2 weeks are focused on protein design. So after we kind of get you familiarized with some fundamentals in the lab, we're going to do a real deep dive into the latest machine learning tools, different types of technologies for designing proteins. And then, so we've got some really wonderful lectures from Xu Guang and Thras, who've been a part of How to Grow since the very beginning. Pranam, who actually was formerly a How to Grow TA, also part of Joe's group as a PhD student, now professor at Duke, and some other special guests will be lecturing as well. And so this will be a big two in silico weeks, a lot of design skills, all related to the protein world. The next 2 weeks, we'll go back into the DNA world and focus on genetic circuits. So really grateful to have Ron Weiss. He's the director of the Synthetic Biology Center. Him and some other special guests are going to be a part of a 2-week set of courses all about the details of genetic circuits, how do we build them and use them for interesting applications. We next have a class on cell-free systems. So everything that we'll have shown you up to this point is primarily working in bacteria. But with cell-free systems, we'll get into the world of working with what are called cell lysates. So basically the guts of cells. And you can do really amazing things with them. You can also incorporate them into textiles, which is a big use case, I think, for some of the designers and artists that are in the room. You can also explore things like synthetic minimal cells and also look at different types of education applications. So we'll dive into all of that in week 7. The next week, we will focus on biological production. So how do we bring together all of these worlds of a DNA design, but specifically around producing molecules of interest? This is a big kind of metabolic engineering / biological production week. And again, there'll be a lab connected to that as well. The following week, we go into bigger constructs. So again, we kind of start with individual proteins, individual genes. We move up in complexity to genetic circuits. And then in week 9, we go into the work of building with whole genomes. And so George, John, and Jeff, the 3 kind of, GJ faculty are really, truly the world leaders and pioneers in how to build and work with big DNA. So really excited for them to be here. And the final sets of weeks of the class, we're going to cover a whole bunch of different topics. So we always have a wildcard week in How to Grow. And so we feature different speakers, including topics around astrobiology and synthetic biology in space, tissue engineering, engineered living materials, RNA technologies, working with other types of biological materials like mycelium or lab-grown materials, DNA sequencing, nanostructures, and more. So we'll have at least one wildcard week. We're also going to have a class on advanced measurement and imaging technologies. So no matter what you do in synthetic biology, you need to be able to measure. So we'll do some fundamentals and explore some of the cutting edge tools there. Joe's already mentioned kind of multicellular systems. One of the most interesting ones that is currently in the room right now is our microbiome. We've got trillions of microorganisms that are part of the human body. We'll talk about that. We'll also talk about frugal science. One of my grad school mates, Manu Prakash, who's now a professor at Stanford, Jenny Malloy at the University of Cambridge, myself, will talk about the world of frugal science and open reagents and microfluidics. And then again, I'm really, really grateful to have Suzanne Lee and Christina Agapakis, who are 2 of the real prominent biodesigners historically, who are going to be here and lead a whole course on biodesign and fabrication. So that's kind of the structure of our lectures. It's going to be an action-packed set of weeks. We're also going to have a topic on eco-augmentation. I forgot. So myself and Bridget Baumgartner, former DARPA program manager and former program manager at Revive and Restore, we'll talk about ecosystem level technologies for synthetic biology. The very last class is going to be you all. Okay, so as George mentioned at the very beginning, one of the really cool parts about How to Grow, each week you're going to be learning skills in the lab, and all of this is going to culminate towards a final project. And we will kind of time travel, 14, 15 weeks. Each of you will be presenting and sharing your project, and this goes also for our global students who are committed listeners. And so, last year we had like 9 hours of final presentations. It was epic over 2 days. You can see the videos online. It's really, really amazing. And just to give you a sense of what some of these projects have looked like over the years, this is one from one of our students, Pat, who is making a microfluidic wearable using optogenetics to trigger the production of different molecules that could be useful in space exploration. Part of what was cool about this project, this was a How to Grow project that turned into a thesis. And this is one of the things, again, just to emphasize for all of you. I should have mentioned this as well. Each of you is going to have a student portfolio. So every single week, you're going to document your progress related to each of the labs, what worked, what didn't. And that portfolio is something that you'll have with you for the rest of your careers that you can point back to. And the final projects are also something that you can all use as a building block for something that you care about. This could be your thesis. It could be maybe a company you want to start, a social good project, whatever it might be. And this is really where your passions and interests really can guide that. So this is a really neat example. This is another project from Al, who is a head TA in How to Grow for many years, which turned into both his master's thesis and I think also a big part of his PhD thesis currently is related to his How to Grow project. This is another project from one of our global students, Yishao, who again had never been in a wet lab before and became a CEO of her own biotech company and was building desktop biolabs. This is a great project from Belend, who is, again, a management scientist or a management specialist working in Sloan, and collaborated with one of our faculty, John Glass, on making a whole cell, a COVID-19 biosensor. So really amazing projects over the years. This is another neat one from Laura Gonzalez showing these architectural shading structures. Again, many of you I know are in architecture at the GSD. So it's a really great opportunity for you to explore something at the interface of one of your technical creative interests and then biology. These are some other really neat projects, screen printing fabrics, textiles that are incorporating engineered organisms responsive to temperature and pressure. This is a really neat one from Andreas Rico, who's a city scientist. He built a series of bioreactors that can be scaled in cities for biological sensing, computational enzymes for breaking down plastics, spider silk spacesuits, really kind of whatever you can imagine is something. This is your opportunity in the final project for you to express it. These are some final projects from this past year. Again, some of the students from last year's class are gonna share their experiences a little bit later, so I won't echo their experiences now. But you can again get a sense of some of the different types of projects, from materials to computationally designed enzymes, artistic projects, and beyond. So again, if you go to the How to Grow website, you can check out and look at all the student projects in past years to get some inspiration. But this is a really, really big part of, I think, why this class is exciting, is your chance to explore your different types of creative pursuits through Synbio. So, all right. And now let's get through some course logistics, and then we'll pause for some Q&A, and then we'll take a break. All right. So if you're going to take a picture of one thing for MIT and Harvard students, take a picture of this screen right here. I'm going to send an email with all this information as well after the class. But this is the course webpage, 2025.htga.org. Okay, so this is this year's course website. And again, we've got a really packed room full of students. Unfortunately, we only have around space in the wet lab for around 20 to 25 students in the wet lab. Okay, so there will be a waiting list, but unfortunately, we're not going to be able to accept all of the students. So critically, to basically be considered for one of the slots, it's super important that you do the homework this week, okay? You are all here in this class, which is one of the other elements, but we need you to come in person to recitation tomorrow. It's going to be at 5 p.m. in room 359, which is around the corner. And then basically do this week's homework, which we're going to talk about in detail in a moment, all right? But the flow of class every week, as I mentioned, first hour, homework review, second 2 hours is lecture. Wednesdays is our recitation. And again, Suvin and a combination of in-person TAs will lead the recitation. That's where we'll go over the homework, what's going to happen, and then potentially even start with the wet lab work on Wednesday. Okay. Each week is going to be a little bit different in terms of the wet lab demands. Typically, we'll have some type of wet lab Thursday and Friday. The hours are a little bit flexible. So I know all of you have courses and other things going on. So this is something as we work through this semester, we'll kind of figure out and get a schedule going. But typically, we do in-person lab hours Thursdays and Fridays, sometimes over the weekend. And then this email address, especially this week, is really important. If you have any questions about the homework, just email this mailing list. This is all of the MIT Harvard TAs on it, MIT and Harvard TAs on it. So email us if you guys have any question. And then the other key thing is once you're actually accepted into the course, we have a whole Slack workspace where you all will be able to interact with us asynchronously, and along with also the global students. So it's a really neat kind of a larger learning family that we kind of build out. So most of the wet lab stuff is actually going to happen in the biology teaching lab, which is diagonally across the street. We also have a wet lab space on the 4th floor where some of the media lab, some of the liquid handling robot work will happen here. But basically, you're going to be primarily across the street in the biology teaching lab. And I really want to shout out Vanessa and Anthony from the biology department. They are so amazing and have been working with us for years now on How to Grow and making it happen. So, all right. So as I mentioned, okay, and this is really important, and I'll pause for questions in a second after this. If you are moved to be considered for one of our slots, come to recitation tomorrow. And your homework is as follows, okay? There's going to be safety training. That's the big thing that's going to happen tomorrow in the wet lab. Susan's going to take you in the wet lab. You guys are going to do safety training. There's also an ethics homework. So I'm going to give a lecture in the second part of class. There's a homework all about governance and ethics, okay? So that's going to be a kind of a design homework that everybody can work on, both students around the world and also MIT and Harvard students. And that's going to be, and also there's an optional kind of lab fundamentals for those of you that have never been in a wet lab, never held a pipetter before. Thursday, we'll be doing some kind of in-person work on how to pipette, how to do very fundamental lab stuff. So that's all this week. And then the other major part is that you're going to set up your own student webpage, okay, where you're going to share your first homework. And so the beginning of class a week from, if you time travel a week from now, those of you that both come to the recitation and do all of the homework, all of you will be considered for one of the slots, and hopefully we'll be able to take everybody. There will be a waiting list, but hopefully we'll be able to take everyone at that stage. So that's basically the kind of plan for this week. So principles and practices homework tomorrow, training on how to build a website, and then the safety training. I'll talk about group projects in a minute, but we've already talked about the individual projects. One thing to mention here, and again, you're gonna go through a tutorial tomorrow. Your individual Notion page, as I mentioned, this is your portfolio. This is your place to share everything that you've done every week, what worked, what didn't. It's basically your lab notebook and your way to present weekly what you've been doing as a part of your work. And the other thing we do is during the first hour of class, we have like a little a wheel of unnatural selection where a group of you will have to present your homework. So it's our way of kind of ensuring that you all will make sure you do your homework, and a group of you will have to present as a part of that first hour. Grading, a third's going to be on this homework. A third's going to be on attendance and participation. So we really, really want to make sure that you're engaging with us fully, both in person and also in the lab. And then finally, your final projects will be the other part of your grade. For listeners, okay, and I'm going to spend more time at the very end of class, so please hang around. You're basically two levels at which, for the students around the world, you can engage with How to Grow. You can be a listener and just pop into lectures whenever you want, but the other part is being what's called a committed listener, okay? And for committed listeners, this year, for the very first time, we have 8 sites around the world where we have TAs that are present. And for those of you that are students in the class, in future years, after you finish and graduate and take this course, you also can be a TA and also help and be a part of our global learning community. So our 80 TAs are distributed also across these global nodes. And so for committed listeners, the students that are going to be doing all of the homework alongside our MIT and Harvard students, you will be assigned to one of these nodes. You'll have TA support. You'll be a part of the Slack workspace, and then also some WhatsApp communications. And the whole goal for our global listeners, committed listeners, will be to actually work and do both the individual final projects and the design homeworks every week. And so for our committed listeners, really important for you all is to do your best to attend class. I understand there's time zone issues for a lot of you around the world. So if you can't come to recitation, do your best. But the most important thing is to do the homework, okay? Make sure you do your homework and have that your student page submitted. And that will help us at the beginning of next week, again, decide the final cohort for our listener community. And so just for those of you that are listeners, we have certificates that'll be signed by myself and George and Joe for everybody that completes all the homework and does all of the homework assignments and the projects. And this is just from our graduation with our global students last year. As an example, it's our first time we did a graduation, which was really neat. And now I'll just very, very briefly have some of the folks here share about their nodes. So is Sally here? Or somebody from GenSpace here? If not, I'll just point out that GenSpace in New York City is one of our nodes. So they will be hosting virtual students and also in-person students if you're in the greater New York area. We've also got Celsius in San Francisco. They are another How to Grow node. I don't know if Elliot is here or if any of the other folks from Celsius are here. But if not, that's fine. So again, great to have an official safe space in San Francisco. So if you're in the Bay Area, you can join in person. But also if you're in the same time zone in the West Coast, please do join our teaching cohort there. Victoria Makerspace, I think Derek is on an airplane right now, but I'm very, very excited to have Derek Jacoby and his team as a part of folks that are in Victoria, Canada, and again, also in that time zone. Ottawa Biosciences, Adrian, are you there?
Yes, I'm here.
Adrian, and if somebody can spotlight Adrian really quickly. Okay, Adrian, yeah, go ahead.
Okay, so I'm Adrian in Ottawa, Canada. This is an EDT time zone. Together with Daniela and other global TAs, will help you navigate with this course. We put together a node website that contains recitations, office hours, protocol information. We will be hosting office hours, recitations, and several labs on Wednesdays, Thursdays, and Saturdays. I did a lot of homework review, so I'm very familiar with that. I would like to know that we welcome you regardless of your geographical location or knowledge in the domain. And we're going to help with homework, reviews, better understanding of lectures and labs and the final projects. We know that many of you don't have an access to the labs, so we'll try to video some of the labs, and maybe we're going to discuss the techniques and outcomes and alternatives. I also organize.
So Adrian, I'm gonna stop you there, Adrian. Sorry, but that's perfect. Thank you so much, Adrian. And for folks that are in that time zone, again, it'd be really, really great to have you there. And then we've got Quito in Ecuador. Ben, are you there?
Yeah, hi everyone. It's real pleasure for us to have the global node for Latin America here in Ecuador this year. So we'll be having hosting the homework reviews, global recitations. We will be having the global open trans laboratories, and also we'll be having some global practices with some students inside of the area, inside of Quito, inside of Ecuador. If you know it, if you are here, please write us. We will be having these sessions, but for sure this will be also for the community listeners. So happy to be here, and yeah, everybody in the area. So please feel free to join.
Great, thank you, Ben. And then UK, Danny and Digby, and if somebody can spotlight them, that'd be great. And just 1 minute to you guys, go ahead.
Yeah, welcome to everyone. Europe's node is LifeFabs, so we're a biofabrication lab and research center. So sort of cross-discipline and sort of symbio, biodesign, and biomedical science. We sort of offer research space. We'll be guiding you through the course, through sort of homework hours, open trans labs, and in-person labs as well. Looking forward to it.
Thank you. Awesome. Thank you so much, Digby and Danny. And then BioClub Tokyo, Georg, are you there?
Hi, everyone. This is Georg. I'm from BioClub Tokyo. So this is very important for people in Asia or Australia, in the Asian or Oceania time zones. So we do class and homework reviews Mondays and Thursdays at 9 in the evening Japan time. So please come and join us. Everyone else is, of course, welcome as well. And this picture is of you, David, 10 years ago when you visited us.
Oh, that's yeah, there we go. There it is. Awesome. We love BioClub Tokyo, and Kat, great to have you here as well. Wonderful.
Yay,
and I apologize, Patrick. I copied everybody's slides in a little bit earlier. So we have Patrick here, who is representing, but again, former TA last year and is now also going to be leading a node in Korea. So you want to quickly, Patrick, you can introduce yourself as well.
So we're starting a node in South Korea, in Incheon, actually, the Yonsei University International Campus. It's going to be the first and only node in South Korea, but we'll be sharing some resources in Zoom review sessions with BioClub Tokyo, as I understand. So then we'll have the similar time zones, and there's some people, and we have our own global TA. One of our students, Kanseo Kwon, is going to be our lead TA for the node. We're going to have in-person lab sessions as well as open trans access. But please contact us. I hope some of these slides get shared because the website and emails are going to be there. Please contact us to make an appointment. Right now, it's only for Yonsei students, but hopefully we'll be expanding to accept community members as well.
Okay, wonderful. And again, thank you all so much. So just to say, I think one of the reasons why globally and for our MIT Harvard students, this was a paper we published in Nature Biotech 2 years ago now. And during the pandemic, we had a situation where we had this hands-on synthetic biology course, but students could not be in wet labs. And so what we did was, and this is why the laboratory automation has really been emerging as a big theme for us in the class, we taught our students how to program robots, okay? And again, I think this is going to be a huge part in general of where we go with synthetic biology. I think there always will be a part, especially for folks that are working with materials. There's going to be an aspect that may require certain types of hands-on work, but increasingly, especially for liquid handling, I think we're going to see a lot more use of robotics, and being able to program robots is going to be a critical part. And it's also been really exciting because for distance learning, this has always been one of the big challenges. How do you teach at a distance when you have to do things hands-on, and mediating through liquid handling robots, it turns out, has been a really big and effective way. And one of the things too is all of the students here and TAs, you all will have an opportunity, if you're interested and motivated, to co-author research papers with us. So this Nature paper was co-authored by students from the course, the vast majority of whom had never been in a wet lab before. So it's pretty amazing. You come and take a class like this, and then a year and a half later, you're a co-author on a Nature biotech paper. So we'll have a similar opportunity for you all as we are building right now the data sets for our follow-up to this paper right now. So this is all part of a larger vision. All of the nodes that you saw, there's a standardized set of robotics that are present at those nodes. And this is something, we've got up to 35 sites around the world that are we're now continuing to scale to beyond this initial group. And all of this for us, I think, is this larger vision around robotics-enabled transformative learning through synthetic biology. You could be anywhere with a computer and an internet connection and engage with our larger network. And so this is something that we're really excited about. And if you think about it, there's a couple of abstraction higher layers here as well. There's a hardware layer with robotics both at MIT and also globally, but then protocols. And you're going to learn all about different protocols, robotic protocols that represent different experimental workflows. And these can be shared. These can be remixed. So colleagues in London can send a set of protocols that here at MIT, we've got standardized robots. So there's a really interesting set of research that we want to explore, both for reproducibility, but also being able to remix different types of ideas that can happen globally. And, again, for those of you that have worked with Scratch as an example, I think there's a lot of really exciting opportunities for design interfaces. Like, what does the Scratch of synthetic biology look like as it relates to protocols and connecting to this robotic network? So this is all kind of this larger context for you all as we move forward. So I'm going to turn it over to Suvin now to talk a little bit about the weekly homeworks. Actually, before I do that, because I've been talking for a very, very long time, I'll pause now for questions while I get this set up for Suvin. Any questions from our MIT Harvard students about anything I've shared so far? I've been talking very fast and a lot. So questions? Yes. Great question. It's the last class. I think it's May 12th or 13th. And for the, they're back to back. So it's going to be it's going to be the second Tuesday in May. And then for our global students, we do like a 6-hour session, however long it ends up being, the following Wednesday. So it'll be Tuesday and then Wednesday back to back. Other questions? Yes. Yes. So it should just be at 5 o'clock. So come to room 359 at 5 p.m., which is around the corner. I'll send out an email tonight with the detail as well. But 5 p.m. in 359, and Suvin in a moment we'll say a little bit more about what we're going to do. Yes. Yeah, so
I'll chat more about that. I think it really depends on which lab we're doing, but it's generally going to be Thursdays and Fridays afternoon. So if you guys want the number right now, it's around 1 to 4 p.m. We can, if you need to come in a little bit later, that's totally fine. Not all the labs will take the full amount of time. Some labs might require to wait for like 36 hours or so for maybe a transformation of E. coli. So it really depends, right? So generally, I would block out your calendar for Thursdays and Fridays, 1 to 4 p.m. as a starter, and then we can go from there.
If you have a conflict though, we still may be able to work. Reach out to Suvin and we may be able to work around your conflict. So, but in general, lab times are on Thursday and Friday. Other questions? Yes, go ahead. So it depends on the lab. Like, for example, this week is really only for folks that are brand new to the wet lab. If you've been in a wet lab, if you know how to do kind of the basics of bio, you don't need to come in person for this week as an example. But some weeks are a lot more intensive. And actually in a moment, Suvin's going to talk a little bit more about some of the details of the lab modules. And so some of them are very intensive weeks. Some of them are a little bit more relaxed.
Yes.
Yeah.
And we'll also share that too. Ahead of time, we'll share this PowerPoint and I'll go and add the estimated time amount. So that way you guys are aware in the beginning of the semester. Again, all of our protocols for all of our exciting labs are going to be up ahead of time. So you guys can see the entire course, be able to know when these labs are going to occur, and then see what we're covering. But yeah. It's on demand. We can chat about that if you're if there are specific conflicts, let's say you're going to a conference some weekend, just let us know. We'll try to work out an arrangement. And we have makeup times at the end of the semester too. So around early May.
Yeah, so part of what's going to happen is the first, you're going to be doing, weekly skills development for the first around like 8 to 9 weeks. And then the last month plus will be a little bit of homework, but mostly devoted to your final projects. So if you missed a module, you can also do makeup time towards the end. Obviously, we want you to be learning, you'll learn the best with your peers the week that the lab module is happening, but there is a little bit of flexibility there. Any other questions? And again, we're going to take a break in like 10 minutes. We're so close to a break. You all are doing wonderfully. If there are no more questions for now, Stephen, why don't you speed through this?
Sounds good.
But this is a set of just details about the lab modules. Hey, global students, are you all seeing slides in full screen?
Yes.
Yes.
Okay, perfect. All right, I'll advance for you, Stephen, so you can speak.
So again, synthetic biology is a very vast field, so we're not going to be able to cover everything. Each one of these topics is pretty much going to be like it can be its own PhD level work. So, however, the course itself aims to like go through the fundamentals of central dogma, again, like as David and Joe described. So each of these weeks are really key towards helping you experience and get accustomed to the wet lab traditions, and then build your foundations on how to invent and innovate in this field. So the first week, this week, if you want to check out the lab, and we'll be covering safety and liquid handling fundamentals. So specifically, if you've never walked in a wet lab, don't worry. If you also didn't understand the terms today, don't worry as well. We'll cover everything. I'm sure it'll be fine. So this week we'll cover bio and chemical safety. This is mandatory for tomorrow. We'll also cover some of the fundamentals for like pipetting and stuff. Week 2 is going to be when we start manipulating DNA. First, how do we visualize the size of it? How do we use different tools online to go and change the structure and make what we want in this? And we'll also show you some databases that exist to help you get accustomed to this at a time. We'll also teach you some of the key proteins, such as restriction enzymes, and how they can be used to adjust and actually cut DNA like scissors, which is pretty exciting, and then actually go ahead and image it.
And just to mention too, this is kind of the first 4 weeks are like protein work. So you're going to be working basically on in the individual genes, DNA manipulation of individual gene length fragments, and then design related to those.
Again, I really want to shout out all the TAs in this room. They're all going to be making it possible. So each of these labs, we're going to be rotating between 2 to 3 different TAs that'll be helped join throughout the semester. So every one of these, we're going to have a ton of different people available. So week 3, which is really exciting, was alluded to earlier, is our Opentrons week, where you get to see the exciting opportunities of lab automation and these liquid handling. So basically a pipette, but on steroids. So you get to use, and a lot of these skills are going to be required for some of these labs that you can see ahead of time. For example, you'll be using Python, Jupyter notebooks, which if you don't recall or like haven't used in a while, feel free to check out our boot camp. So a lot of these tools are also available online again, and we'll be going through making wonderful art, but also exploring the fundamentals of bio with like bacterial cell culture. So next slide. Then we're going to use a lot of these in silico tools we discussed earlier. So for example, AlphaFold, Rosetta, to go model a lot of these protein folding that has been really awesome the last few years. So a lot of these lab skills are going to be involved in like such as like aligning these proteins and being able to visualize them and optimize them for how well they perform in actual experiments. Because for example, these are very difficult models to compute. And so what's really exciting, you can go ahead and do this in the course of 2 weeks. We'll also have this tied to a group final project that you can use to build off your work and create it. So for a lot of the ECS folks and CS folks here, it's going to be wonderful. So they'll be able to explore this. And this might be up your alleyway. So feel free to look forward to that. This is one of our capstone labs, and honestly, one of my favorites. This lab allows you to change the outer protein of E. coli to express as many colors as you think. So you can change from purple to pink to blue to magenta. So a lot of these really take into account the skills of we've building up so far. So for example, you can probably hear a little bit of excitement because you've been working on DNA design, the central dogma to really go ahead and actually edit and adjust this DNA and assemble it together. And so this lab happens in the middle of the semester. And once you get through this lab, the rest of the course will become a piece of cake and you can build there with your exciting projects. So we'll cover how to image it and how to measure it as well. New to this semester is a neuromorphic circuit. So we'll also focus on genetic circuits. So again, we have 2 weeks on those lectures. And sorry, I am speaking a little bit fast, I apologize. But
I'll cover this in a little bit greater detail, the genetic circuit lab.
So we'll be covering this, and we'll be working in lab. So all the experiments I described again, except for the in silico ones, will be happening in lab. You'll get to use your skills there for able to do it in real time. And so this will just build on top of that. The next week is going to be focusing on cell-free, so specifically focusing on protein design, expression, and purification. But what do you do to like generate these kinds of proteins without the required need of a chassis, which in this case is coli, so the organism that we're using. And also, this again is a very PhD level topic that we're going to only scratch the surface of. So a lot of these topics again are really exciting, but will take time. Bioproduction is another one of our cool labs where we get to see how we can use these E. coli to manufacture our biomaterials and biomolecules that we can use, for example, for pharmaceuticals companies, et cetera. So we'll be experimenting with different E. coli cultures of how to grow these in optimal conditions and then go ahead and measure. So I think you might be starting to see, if you hope you aren't too tired yet, like a recurring theme where it's based on editing DNA, assembling the DNA, and then being able to actually go and visualize what's actually happening. A lot of this is very tricky in synthetic biology, and it's been really made possible in the last few years to do so. One of our final labs is the CRISPR lab. We'll be able to use an engineered version of CRISPR-Cas9 with a very intricate but fun 3 plasmid design to edit GFP, which is the fluorescent proteins that you might have seen on the news and so forth. And one of the cool additions, which Bren is helping here in a little bit, is week 11, which is about living materials. And so we'll be using this lab to go ahead and create 3D living materials made from bacterial biocement, which will be a very exciting time to apply a lot of our course into different applications. So I'm not doing the topic justice here explaining it, but all of these topics here so far are going to be like a fraction of field itself and really exciting ways to engage with the curriculum. I think these labs range from 3 to 6 hours, depending on how much time it takes for transformation between them. But again, the exciting part of the in-person labs is that you get to go do this and get yourself. You can see the result of the color change, that your edits made this happen. And these skills will hopefully help you prepare to not only become better scientists, but also be able to go and actually enable your activity in this field in real time as a researcher moving forward. So a quick introduction, and again, don't worry if this is confusing. Don't worry if you didn't understand any of the words. I was in your same boat. It's okay. As a freshman, I remember I was like, oh my God, what is this even? Like, what is the central dogma? Embarrassingly, I asked that question here, but so I think it'll be exciting to like go through.
Let's wrap up. Yeah, perfect. Okay, thank you. And let's just big round of applause to Subin and all the TAs. There's so much work that goes into preparing all this. It just, we've been meeting, really, this class actually doesn't stop. We meet continuously throughout the year about it. So I'm really grateful to all the TAs. And this is Subin, and you're going to have to take us on one of your flights sometime. All right. So lastly, and then we're going to take a break. So one of the really kind of neat parts about How to Grow the past 2 years has been working together on a collaborative project. So if those of you that are familiar with IGEM, the International Genetically Engineered Machines competition, that's like a competition where teams work and compete against each other. We are trying to establish a type of model where we actually can all work together on projects globally with a common goal. And so very quickly, I'll give you an example from last year, which we're continuing on this year. So one of the major global emerging health threats is around antimicrobially resistant bacteria. So one of the kind of solutions to this are these wonderful little bacteriophages that can be designed to infect these resistant bacteria and can potentially act as a therapeutic. And so what students did last year, and you also will have an opportunity to do this year, is to design your own lysis proteins. Okay, so and there's, again, this will be a big part of the protein design class. How do we optimize towards certain desired traits that we want from these lysis proteins? But the goal here was to have students at MIT and Harvard, and also all around the world, try out and have different protein designs. We would then have Twist Biosciences, which is a biotech company that, again, we work with that does DNA synthesis. They'll synthesize those DNA constructs, and then we'll test those DNA constructs and express those proteins, both using digital microfluidics, but then also via this network of robots at the nodes that we were just talking about. And so just to give you a sense, this is actually from last year's class. This is a little video of the digital microfluidics. It's a really neat technology. Each one of these pixels represents a different student protein design. And so they're all being mixed with the appropriate reagents and then expressed on this device, meaning the protein is actually synthesized in this device and also tested. So, so this is something that we did in person here at MIT. But a big part of what will happen globally is both expressing the proteins and also testing their efficacy in breaking down bacterial plaques. So this is just some data that was from our students last year, and a good bunch of the student designs had improved folding compared to the wild type. And again, as I mentioned, this year, we're going to be scaling this. And for our global students, you'll also have an opportunity to try and assemble some of these proteins locally at the various nodes around the world. And lastly, so this is a part of the experimental work. So we got a bunch of data last year. We're going to collect more data this year, and this all will inform, hopefully another big group paper that will be a follow-up to our Nature Biotech paper. And lastly, there's another group final project that we're doing in collaboration with Ron Weiss. Again, he's the director of the Synthetic Biology Center around what are called neuromorphic circuits. All right, so we're going to talk a lot about these genetic circuits, and there's a particular type of circuit that can emulate neural network behavior, but in a cell. All right, and so part of what is really neat about this is that you all will have the opportunity to design your own neuromorphic circuits. So there's a design environment in which you can, in silico with your computer, everybody around the world can work on designing different types of neuromorphic circuits. And then we're going to use robotics to actually build different combinations of the circuits. We'll do that both at MIT and also globally using a set of parts. And so this will be like a big kind of DNA assembly project. And, part of the goal here, which you can imagine, we've got one project on phage therapy, this genetic circuit project. What we really hope to do over time is to have this global network of students and nodes working together on different types of projects. So this is one where we're all going to build different DNA constructs and test them. You can imagine testing them for reproducibility. You can pick top designs that are happening around the world and then iterate collectively based on those outputs. So there's a tremendous amount that we can do with this larger network as we build it out. So we're really excited about that. And I'm going to stop now since we're exactly 90 minutes, which is amazing timing wise. So, and we'll try to figure out what's going on with the visual, but why don't we take a 10-minute break? Again, you all have been heroic in lasting for 90 straight minutes through this, but we're going to take a 10-minute break right now, come back at 3:40 p.m. Eastern time, and we will complete the last stretch of class. So thank you all so much, and we will be back. All right, everyone. So why don't we get back started again? I'm very excited for this part of the class. This is kind of a tradition that we started, I think last year. Okay. So we're now actually going to have a few of the students from last year's class share about their experiences, a little bit about their projects, and then just their experience and how to grow and how that's impacted them in their academic careers and lives. So I'm going to turn it over. We're going to have four student speakers now. And so I'm going to turn it over first to Lenny. So actually, if everybody give a round of applause for Lenny Jefferson. Lenny.
Thanks, David. Yeah, it's good to be back. So I'll start just going over sort of what my final project ended up being, and then pitch you why I think this class was really good for me, and that maybe that you are in a shared situation. So I was sort of inspired in the third module, the OpenTrons module, when we were doing this kind of gel art, which you can see here on the left, including like a simulator module and then printing sort of bacteria culture on this plate. And I used ChatGPT sort of, to specify a design for a kind of, obscure Python package, PyLab robot that my lab develops to, make lab automation easier. And it one-shotted it. And so I thought, maybe this can actually be extended to kind of remove even the programming barrier to lab automation. So if you can instead specify, both simple commands, but also protocols in natural language that could make lab automation quite a bit more accessible. And so that was sort of the concept that we went with for my final project. And I collaborated with another person in the class to kind of make that happen. And we basically build kind of an agent that can take in natural language and, write the code that you can then execute on an OpenTrons or any other liquid handling robot. So that agent exists and we've presented it. And that work is still ongoing. But I think, more broadly in terms of what the class offered me, I work in a sort of pandemic preparedness, infectious disease biology group. And I was have always been on the sort of dry lab or computational side throughout my kind of research career. And so this class was a really good opportunity for me to kind of see the wet lab side and kind of just go through some of the, I don't even want to say basic because you really do fairly sophisticated, labs at the end. but just like doing primer design, using benchling, right? It gave me kind of a way to understand and communicate with the wet lab side of the lab much better. And it's something that I now feel kind of empowered as like, okay, if I do, choose to do some wet lab work as part of my thesis, then that no longer feels daunting, as it used to. I think beyond that, even the sort of community that How to Grow connected me to, both as like physical, my sort of classmates in that cohort, but also like the just amazing group of speakers that we had here and the sort of ability to engage with them later. So Drew Endy, who's a speaker, he ended up being on my master's thesis committee, and that was sort of someone I met through this course. I visited a couple other How to Grow friends in at Life Labs London. So that was a connection that I had made through this. So I generally feel kind of more connected as part of like a global DIY bio community. And I think that's something that I wasn't really aware of existed. And now I feels like, I have connections, kind of all over the world to hack and talk biology if we want. So that's the very exciting thing that I'm very appreciative of this course for. So I'll leave it at that, but yeah, I think you should take it.
Lenny, awesome. Thank you so much. Great. All right. So up next, and Lenny, I think is a great example of somebody who has a lot of computational expertise and again was able to get some wet lab experience. I know many of you are also in the design world, and so Ya, who is a designer, also is at the GSD, where a number of you I know are doing work, is going to share about her experience. So yeah, go for it.
So hello everyone, my name is Ya. So as David just mentioned, I have a background in consumer product design. So as you can tell, like I'm a totally new beginner to biology. So here I kind of want to share like a perspective as like a new beginner to biology. So why I chose this class for me personally is because I'm interested in like how can we manufacture products in a more sustainable way. So like biomaterials sort of like the natural thing that comes to me, and I just feel like it could be like sort of the disruptive technologies that could be the future of things. So that's like my reason of being here. So, but I'm sure like each one of us must have our like own unique kind of reasons of why being here. So I guess I'll just jump right into my final project, which is creating this sort of like fluorescent bacteria cellulose to be this kind of like natural laser-ish material. This is sort of like a rendering that is that I made. So this project, it's kind of speculates on designers' ability to promote acceptance of biomaterials among mass consumer spec biologically synthesized bacteria to develop visually captivating materials challenging the traditional take-make-based product development process. I won't dive too much into this thing, but this is sort of the thing that if you take this class, you would also do, which is to come up like the 3 aims that your designs are gonna be, such as like the first aim is it's sort of like just like the basic level of like what you're gonna do, but you're gonna look beyond the first thing, which is also like I feel that's the biggest takeaway for me is to learn this new world of synthetic biology and to connect design, synthetic biology, and technology. Here are just like some things that I won't like spend too much on this part, but you're gonna get a lot of like hands-on experiences, like the resources here, like MIT, it's like it's a lot, like that's very helpful. So I guess like the last thing I would want to say is like I think one of the most charming things about this class is actually like the people around you, like they just here, like your classmates, like all the TAs and professors. So that's definitely like why you be in this class. So yeah, thank you.
Thank you, Ya. All right, Hugo.
Thank you, David. Yeah. Hello, everyone. My name is Hugo Zhang, and I'm a first-year PhD student at the Media Lab. And I'm here to share my experience on last year's How to Grow class and about my final project briefly. So before moving on, I just want to I just want to share my background because I think many of you will think about I'm not a biology background or bio-related major. So can we do well in this class? So I'm also electrical engineering background and material science background also. And I usually make a PCB design or wearable devices or characterization and troubleshooting, just the typical things that electrical engineers do. But I hope you guys will. Thank you. I hope you guys will do well in this class since I also went through well, and I was really impressed in this class by two things about the classes and the lab classes. For the classes, I could hear I could hear from the world leaders of each field, and they can explain all the key concepts or complex concepts with a really easy words, so any beginners can understand what they need to know. And also the lab class is kind of the highlight of this how to work class. So David or Subin will teach the almost every essential fundamental lab techniques that you need to do in synthetic biolab. So after this class, I'm keeping synthetic biology after this class, and my PhD project is also a synthetic biology. But from this experience, I learned a lot, and it helped me a lot still. And yeah. So my final project was really simple. Like I wanted to make a cell sensor that can recognize the tumor. So the idea is relatively simple, that you just genetically modify the cells to express the synthetic receptors that can recognize the specific antigen of the tumor. So the idea is relatively simple, but the actual experiment was really hard. So I had to go through like 8 steps to make the whole cell sensor and characterize it. But honestly, I couldn't do this during the semester because of 2 labor-intensive experiments, and I could do only plannings so far. And so my advice is make a realistic plan and always always make a plan ahead of the final projects periods. So although I didn't finish my final project by the end of the semester, I'm keep doing it as my PhD project, and it keeps going on and going on and going on. It never shown me the results, but I hope to see a result soon. But anyway, yeah, I learned a lot from this class, and every knowledge or every lesson that I learned from this class keep helping.
No.
Keep helping me.
Mañana voy a leer.
We have somebody needs to be muted, but anyways, thank you,
Yeah.
TAs. Okay, go ahead, Kyo.
Yeah, keep doing me the research in synthetic biofield. So I hope you also enjoyed this class and fell into the synthetic biology. Thank you.
Awesome, Kyo. Thank you so much. And again, I think, for those of you, there's a really great opportunity for you to connect your projects into theses. That's a really kind of classic pipeline that that happens for a lot of our students. Okay, so to bring us home, and then we'll do a quick Q&A with all the students, is Kevin. So Kevin, just give me 1 second here. I'm going to reopen this. Okay. The never-ending final project. And this is a... Yeah, good. Thanks for sharing the photo as well. Okay, so Kevin, go for it.
Hi, my name is Kevin. I am a second-year master's student at Harvard GSD. So my background is mostly in industrial design and now mostly in HCI. So the entire time I was thinking about, I'm new to biology and thinking about how can I, as a designer, approach biology, and especially with bacteria. And soon, I think throughout the labs, you will soon learn that bacteria works at a different time scale from what people are usually expecting. Like I'm used to sort of electrical systems or 3D printing, that's super like matters of hours, but bacteria sort of goes in terms of days. So I want to create an interface that sort of brings the bacterial speed to sort of human speed. So sort of my guiding question for my final project is similar to how can I, as a user rather than a scientist, touch and manipulate bacterial gene expression? And I just want to share a little bit about how I approached my final project. I try to simplify everything down to exactly how the whole system works and what do I need to edit in order to make my final goal happen. So essentially what I did was,
Can I get a little closer to that?
oh, essentially what I did was, I took one of the labs that you guys will do, which is the Gibson assembly line, where you express a purple chromoprotein. And I decided, okay, how can I express this purple color whenever I want as the sort of manipulator or the device controller. And there's this promoter called DNA-K, which is a thing that responds to heat stress. And I basically took that thing that causes the heat stress and tied it to the purple gene expression. And essentially what we got in the end was a bacteria that reacts to PCB heating that's modulated by the user. That was my final project, which I think maybe one tip for the class is that it's okay to maybe edit a existing lab that we do throughout the course. And I think that's probably also the best approach to make sure that you end up with a project that will actually work. Not saying mine fully worked, but yeah.
Great, awesome, Kevin. Thank you so much. So why don't we, since we've got the four of you here, thank you again for sharing the student perspective. Did any of you from this year's class have any questions for last year's students? Any questions, comments, thoughts? Yeah, please. So the question's around the different planning phases for the projects. Well, I guess one thing I'll share is that early on, like basically, actually starting with your first homework. So as a part of this first homework, we're going to talk a little bit about ethics and governance. So you're already going to start by picking some type of biological application or biotechnology or synthetic biology application that you find interesting. So already, as a part of week one, you'll design a governance framework for whatever something that you care about, which could be your final project idea. So again, if you're interested in textiles, city sensing with biology, genome editing, whatever it might be, you can start thinking about that already in this week's homework. And then about like a month or so in, month and a half in, we're going to start having you do final project crits, where you'll bring ideas for your final project and share them to the entire class. We'll give you feedback. And then somewhere around like halfway or through the year, we'll start assigning you TAs. So again, there's like 80 TAs, both globally and here at MIT. We'll try to find one, if you're interested in, fuel cells, or if you're interested in computational design, whatever, we'll try to pair you with a TA that's got some expertise or interest in that area, or a faculty member. And then you can then start working over the second half of the semester with that TA or faculty member to actually start designing experiments and do some of the work in the lab. And, again, to the students' experience, bio is just slower than, growing things just takes more time. So we try to set up the final project so you'll be able to accomplish at least some set of experimental results over the last month, and then ideally have a larger vision that you can grow towards, for example, into, a thesis or a paper, or in some cases, even, startup companies, which have also come out of the class. So I don't know if there's anything y'all want to add to that.
I think earlier is always the better. So probably you only have 1 month, which makes it only one mistake or two mistakes is really critical for you. Before the end of the semester, so you should order all the materials before beginning that final project.
Yeah, that's a great comment. And again, we're very, very understanding about kind of the challenges around experimentation with biology, and it does take some more time. So yeah, earlier the better. But again, literally starting this week, any kind of initial concepts that you find exciting, you can start exploring as a part of this week's homework. Yeah, please. Yeah. Yes.
Most of the stuff in order. You basically we'll let you guys order, I think, around a month to 2 months earlier than the final projects do. So you can order enzymes, plasmids, you can order different DNA constructs, custom designs for everything, even kits to go and modify this DNA. So we have a ton of kits that you'll see through all of the labs. And I really like Evan's point to add to this is that, again, building on top of the labs you've already developed is a wonderful idea for multiple reasons. One specifically, these labs have gone through years of iteration, even months. All of TAs have actually been working on helping improve these. So they all have custom kits that you need for building these labs, basically putting in different plasmids, different sequences. All of these are available on these labs already, so you can go ahead and use those. So any of those kits that you find are here, and the rest you can order. But the budget is pretty good for like students and specifically. We can't say exactly how much, but there's enough that you'll be able to complete with the final project and create your own custom videos.
And part of this too, as we kind of figure out, like part of what's going to happen is, as we sort of see, there may be a couple of you that are really interested more in like materials. Some of you maybe were interested in some cell-free protein projects. We'll start grouping you a little bit. And so you can also share reagents and kits with students that have similar kind of themes. So we'll, and again, super grateful to Thermo Fisher. Thermo Fisher is like a nation state. I mean, they're so massive, and they're generously giving us a ton of support. It takes a while though to order from them. There's a lot of bureaucracy there. So one of my slides I've not shared is just kind of the be patient, be flexible slide. How to grow. Even though we've been doing this for years, we are still like building this like giant plane as we're flying it, so we've been bringing on, and we're over the course of the semester, we're going to continue to bring on some more partners as well. So there is flexibility, but to Kyuho's point, the earlier you can get DNA designs and figure out things that you want to experiment with, the more likely we can get those things in person for you to actually experiment with.
I just want to add one more thing. Again, don't fret about this process. We'll walk you through every step of the way. We'll walk you through each of the labs and how to use those in parts of it for the final project. One thing I'll also say is that if you come to the lab on Thursday, you'll see some of these new items and equipment we have for the semester and that you guys can be able to use. So if you guys are curious about the kits, even other items that we're using, please do come on Thursday, even if you have prior experience, because we'd love to show you what's available to those. So.
Awesome. Other questions, comments for now? Everybody feeling pretty comfortable overall? Yeah. So, and again, I know there's a lot of new concepts, but just in terms of the logistics, hopefully this is all making sense. And I know for our global students, again, when we wrap up in an hour, just know that I'm going to stay online and answer questions. So we'll do a kind of a impromptu office hours for all the global students as well. So thank you all for your patience. All right, so let us move on to the next part of this. And thanks so much again to you guys. Really, and actually, one more big round of applause to our students. They're all TAs this year as well. So again, thanks so much to you guys. It's a huge effort here in How to Grow. All right, so now what we are going to do, all right, is I'm going to endeavor to give a lecture about principles and practices. All right. So one of the things I'm sorry about for today, one of our colleagues, Megan Palmer, who is a really innovator and pioneer in synthetic biology, unfortunately was not able to join us in person today. Megan has been a part of How to Grow Almost Anything since our very first iteration. Global students, can you all both see me and see these slides?
Yes.
Yes?
Yes.
Okay.
Yes.
Perfect. All right, great. So Megan has been a big part of How to Grow since our very first iteration. And there's a little course history that I can give a chance to go to, but long story short, for those of you, again, I mentioned that I give a little analogy between computer clubhouses and Scratch and the digital fabrication network, the Fab Lab network, and how to make almost anything. How to grow almost anything actually started being taught not at MIT, but to the network of Fab Labs that were part of Neil Gershenfeld's Fab Lab network. And so the first few years of How to Grow, and this is also why we are already connected with BioClub Tokyo as an example, was taught in a grassroots manner to global network. And it wasn't until around right before the pandemic that we actually brought the course back to MIT. And so Megan is a part of our faculty that has been with us since the very beginning. And we've always started How to Grow, our very first class, thinking about principles, practices, and also ethics. And so this is something that, again, I think it's very easy for us to get caught up in the skills and the really exciting projects and what's possible and enabled by these technologies. But each week, we really do want for all of you to be thinking very thoughtfully about why we should be growing almost anything. How do we do it safely? How do we do it in ways that are going to ultimately have enormous public benefit, ideally? And again, we'll talk about this a little bit more, but there's kind of like a half type of doom that we see both with synthetic biology and even artificial intelligence, right? It's like synbio and AI are going to save the world and destroy the world, like both at the same time. And so it's not through kind of coincidence or accident that we can get one or versus the other outcome. And so we'll talk a bunch today about governance and other ways in which we can try to influence outcomes that are going to ultimately have more public benefit and more benefit towards the climate and our planet. So anyways, I'm going to be channeling my best Megan Palmer for today. I've heard Megan give this lecture many, many times. So hopefully I have learned enough to be able to communicate Megan's message to you all here today. And just to say too, these slides are also adapted from a part of Megan's talk last year. So by way of introduction, this is Megan. And so Megan is actually one of my oldest friends at MIT. We both did our PhDs here at MIT at a similar time. And Megan is a real kind of major figure in the larger synthetic biology ecosystem. So, one thing to mention, I shared that paper from Drew ND from 2003. Synthetic biology as a field was in many ways birthed in part, large part, I would say, at MIT. There were many institutions that were involved, but a lot of activity was happening here at MIT in the early 2000s. One of the big institutions that came out of MIT was something called iGEM, which we've talked a little bit about. iGEM, again, was birthed as this exploration of the idea of standardized DNA parts, right? Could you, much like in engineering, mechanical engineering, electrical engineering, work with standardized parts? Could we do a similar thing with DNA-based parts? And so iGEM was established as a global competition, and in many ways was to generate, it was set up so that students around the world could generate parts that could go into a larger registry, which actually exists. If you go to parts.igem.org, you'll see a giant list of DNA parts. There's now many different types of DNA part catalogs that exist, but this all started in the early 2000s, and Megan has been a huge part of iGEM over the past 20 years and has led in particular a whole thrust in iGEM around governance and practices. So not the technical parts, but really thinking about the societal implications of synthetic biology. So iGEM is a really important institution that Megan is a part of. SynBIRC, which then evolved into EBRC, which is another big network, a global network of synthetic biology laboratories. SynBio Leap was a leadership program. I actually was very fortunate to be a part of that leadership program, along with a lot of other folks at the Media Lab. Kevin Nesfelt was part of that program. Actually, Patrick Boyle, who's one of our speakers, also part of the program. Christina Agapagis. So a lot of our faculty were connected to this larger leadership program that Megan helped to establish. So anyways, she's just a really significant figure. Also worked with Revive and Restore, which again, we'll talk a little bit later on about synthetic biology and conservation, eco-augmentation. And Ginkgo Bioworks, which again has had its own challenges over the years, but I think is a really important historical institution in synthetic biology. And now Megan works for ARPA-H, with which those of you are familiar. There's DARPA, which is the Defense Advanced Research Projects Agency. It's the reason we have NASA and the internet and mRNA technologies all through DARPA. There is an ARPA for defense, but also an ARPA for health. And so Megan has recently started a new role as a part of ARPA-H. So that is Megan. And today what we're going to cover is, again, thinking about this big picture of, what's at stake with growing almost anything. This class in particular, we really do embrace overall a larger open source mentality, really being able to share knowledge, but also sharing culture, sharing best practices, right? I think one of the great things about being a part of this larger How to Grow effort, if you're at MIT or Harvard or around the world, is that you're connected to kind of a cultural inertia and a set of norms, which is really important. So we'll talk about the kind of what's at stake for growing good, growing almost anything, challenges, opportunities, grounding examples, and then your responsibility as a part of this larger emerging field, and then the homework that you're going to be doing connected to governance, again, related to a synthetic biology project that you are interested in. So let's start with the big picture. So as I was sharing, one of Megan's favorite activities is collecting different magazine covers from a synthetic biology over the years. And so this is sort of a really, kind of neat example, again, of this half type of doom, right? There's, no hunger, no pollution, no disease, and then, biology's brave new world and the consequences of gene editing. So this classic like exponential technology that's both going to save and destroy the world. And so, one of the big questions is, how do we ensure that the development of biotech is overwhelmingly constructive as opposed to destructive? And, how can ethics and governance play a role in that? Another thing to mention here is that there's many different types of biofutures that are possible. I've shared, and as a part of this, kind of first 2 hours of the class, we've talked a lot, I would say, in industrial ways thinking about biology, utilitarian ways of thinking about biology. But there are also other ways of thinking about biology, ecological ways, decentralized ways, which is something that, again, we in this particular course, we're emphasizing a lot. So this is kind of a neat figure, kind of showing different axes of possible futures, from destructive to constructive, ecological to industrial, concentrated to distributed, right? Drew Endy, who we've mentioned a bunch, he's talked a lot about, DNA synthesizers being in everybody's home. So imagine having like portable manufacturing. Some people are terrified by this idea, right? Some people think that's going to help to destroy the world. So these are all some of the tensions that we hold as we move forward in this field. And again, we really encourage you as we go each week to be thinking about these possible futures. And again, biostrategies, right? This is an example, engineering SARS-CoV-2 using a reverse genetic system. One argument for this type of a paper is we can make better vaccines. And another argument is, what are you guys doing trying to reverse, genetic engineer SARS-CoV-2? Is this just more bioterror and more bioerror? Right. So at each stage, as we navigate and think about these different projects, there's always kind of a question of like, what are the potential outcomes? What's likely to be in service to the public good and in service to the field? And what are things that potentially can promote harm and promote less constructive methods? This is a neat one. Make your own heroin is almost here. One of our colleagues at Stanford and now leading her own company, Christina Smolke, was part of a larger set of efforts around opioid production in synthetic biology. And there's a lot of folks, again, go back to the biological production class. A lot of synthetic biology historically has been about engineering organisms to make stuff, right? Flavors, fragrances, therapeutics, or drugs, right? So, there's a huge suite of capabilities that are possible. And, technology is not neutral, right? We can apply these technologies towards all kinds of different ends. And there's been a lot of discussion over the years about how should we be leveraging both genetic engineering historically and now synthetic biology. There's a real historic meeting that's shown here. This is from a picture from 1975. There's a very famous meeting that happened in Asilomar, which is a conference location in California. And this is when kind of a lot of the first genetic engineering tools were becoming available. And there was a similar set of questions around, what is safe? What should we or should not be doing with these DNA synthesis technologies that are emerging in 1975? A similar meeting in almost 40 years later in 2015, a genome editing convention also grappling with these same questions. And amazingly, next month, there's going to be the 50th anniversary of Asilomars happening. And again, some of the biosafety that Subin's going to be leading you through tomorrow, those biosafety levels were established in part because of that 1975 Asilomar meeting. We're going to be having a 50th anniversary. It's going to be happening in the end of February. Drew, again, is one of the lead organizers of it. I'm also going to be a co-chair at this particular event. I think a number of you may be even going to this event, or at least a few of you are. But it's going to be a really historic and important meeting. And again, is going to be grappling with some of these larger questions around what kind of future do we want with biotech and how are we going to make that real? So we've talked a lot already about applications, right? So we've talked about, materials, agriculture, conservation, and so on. And so as the application space for synthetic biology continues to grow and proliferate, the associated economic impact also is increasing. And so there's been, a lot of estimates about what the future bioeconomy looks like. Is it, 2 trillion? Is it 4 trillion? Whatever it is, it's an increasingly big number that's going to have a increasingly larger economic impact and also political consequences as well. Obviously, we're in a time right now where different types of national powers are facing off, and biology is a part of that larger set of political economic consequences as well. Right now in the United States, there was a recent National Security Commission on Emerging Biotechnology. Many colleagues and members of the faculty were a part of that commission, which was all about, again, determining our futures with synthetic biology. What should it look like? Should there be distributed education? Should we be emphasizing manufacturing? A lot of those types of conversations were happening over the past year. And actually, there's a report that's going to be coming out soon about these commission's findings. It's really interesting in this moment. Obviously, we had a very significant presidential election. So there's going to be, I think, some really interesting questions going forward about how the new administration looks at biotech. But this is all playing out at various scales, in classrooms here at MIT, community labs, academic labs, and then also at the state level. And so when we think about the different challenges and opportunities, we've got these different possible futures that we can imagine. There's a lot of different technical transformations that are driving all of this change. Now we're increasingly, as you've all seen, hopefully throughout the course of today's lecture, there's this kind of blurriness between the digital world and the physical biological world, right? And a lot of this is enabled through synthetic biology, both DNA design, protein design, all of this enabled in silico using increasingly more powerful tools and with ramifications in the physical world, both in terms of the manufacturing, what we can test, what we can synthesize and make. And now we're seeing, and we'll talk about this in the bioproduction course, but also in the automation class, an increasing use of industrial synthetic biology and large-scale laboratory automation, right? When I was doing my PhD many moons ago in Joe's lab, I was working on microfluidics and these chip-based kind of DNA synthesis devices, but a huge amount of my work and a huge amount of work still to this very day is done with pipetters in a lab. And, I think increasingly and industrially, we're moving towards large-scale robotics, which I think is why all of you getting some experience programming robots is really important for this class. But increasingly, we're seeing the role of large-scale automation. And then as we bridge into digital worlds, how artificial intelligence, Lenny, I think, gave a really nice example of natural language processing interfacing with robotics that can actually control biological experiments. So that's, kind of technical, industrial scale change, and then also social scale change, right? I mentioned iGEM. iGEM started very much like this class. It started, I think, back in, early 2000s. There was a classroom not unlike this with a few students, and now has scaled to a really global set of students and programs. Thousands of students participate in iGEM now every year globally. And a lot of the folks that started iGEM have grown up and are the founders and creators of some of the really significant synthetic biology institutions this day. As we've mentioned, Ginkgo Bioworks is one. Bioeconomy is biosecurity. Increasingly as well, there are a lot of institutions, and a lot of this is driven through the pandemic, thinking about how do we do biology safely? How do we screen for sequences? How do we screen even robotic protocols? This is a huge part of kind of the future of the proliferation of this technology is also around containment and safety. George and his lab has done a lot of pioneering work thinking about biocontainment, not just as kind of an ethical or safety area, but actually as a real technical area of innovation. And so for those of you that are interested, again, biosafety, biocontainment is a really, really big area. I mentioned the Asilomar meeting that's happening at the end of February. One of the big themes, there's 6 different themes that are part of that meeting, but one of the big themes is around biocontainment. So it's a really, really important area that requires a lot of technical innovation as well. So for those of you that are curious about this area, that could be a really interesting one for you all to explore. So this is a neat slide that, again, comes from that 2003 study that I referenced at the start of class. This is this idea that as we get better at DNA manipulation, if you don't get better at detection, analysis, and response, of course, the risk will amplify exponentially. If everybody's got DNA synthesizers, but you have no way to detect, analyze, or respond, then of course you're amplifying risk. And so one of the early arguments from the early synthetic biologists was DNA manipulation, read, write, edit, all really important, but we also need to be able to do these other things as well. I think it's actually an interesting question. I'll pose this to Joe and also to George later, if they agree that detection, analysis, and response is also scaled with manipulation. But I think that's a question we can all reflect a little bit on. But as a big part of how we can imagine synthetic biology scaling in a safe way. So another kind of key question as we kind of get into this idea of governance is, who are the actors and how do we agree, right? And, one of the, kind of historical points that Megan loves to address, biological weapons, there was a period where countries and nation states did that. They actually worked on developing biological weapons, and there were actually treaties that were involved globally to say that, hey, we want to restrict that, and we as humanity do not want to work on biological weapons. And so that's an example of a type of agreement that was arrived at globally that has really had profound implications on how we ultimately manifest synthetic biology in the world. We don't work on weapons. There was a period where I used to work at a place called Lincoln Lab at MIT. I actually had a top secret security clearance, which was really interesting for a handful of years. And it was great to be working in an environment where, while there was kind of national security related synthetic biology, it was not about weapons, right? It could be about chemical detection and so on, or mitigation, but it was not about offensive weaponry. And so that is a result of good governance. And so these governance challenges are non-trivial, right? And we've already talked about some of these areas. Professor Esvelt here at the lab has done a lot of work on gene drives, ecosystem engineering, same thing with George and others. These are really complex ethical questions that involve many, many different types of stakeholders and actors. You're talking about governments, community groups, and so on. Brewing drugs, which you talked about, different types of molecules that have public health impacts. What about editing our cells? This is a really, really big area that George and others have developed enabling technologies for. So really, really big ethical questions that come along with this technological development. And also a potential pandemic pathogens, right? There's ongoing research and conversation about even the origins of the COVID-19 pandemic. So there's a lot of these types of issues that are related and connected to effective governance that we really need to be thinking about. There's a really good set of frameworks, and this is a particular one on biotechnology is ethically entangled. This was an ethics of synthetic biology and emerging technologies document. Again, we'll share these slides later, but for those of you that are interested on the policy side, this is a really interesting and historic document. It talked about principled approaches to responsibility, and not just about safety, but also about questions like who benefits, right? If we develop these technologies, is there a clear public benefit? Is there responsible stewardship, right? For a lot of these ecosystem technologies, there's a lot of really important debate about can we or should we release these technologies out into the wild? How do we deliberate and think about this collectively? Should it just be a few smart people get to decide, or should there be more democratic processes to be involved? How do values of justice and fairness play out as we deploy these technologies? So all of these types of conversations are really, I think, are very critical, and we want you to have this lens as we, throughout the semester, introduce you to different types of technical capabilities and really superpowers, right? If we're talking about genome scale engineering, that's a really incredibly powerful set of tools, but then what are the other ethical dimensions that come along with that technology? And there's a lot of folks that have been have attracted different levels of attention. Joe Zahner, who's a biohacker and a DIY bio, in many ways, innovator, but also provocateur. George himself has been involved in a lot of different projects that have had different types of media attention. And so, all of this different types of attention raises these different ethical questions as well that connect with, how we govern and think about the future of the field. And so, again, rewind 20 years ago when synthetic biology, these early institutions hadn't existed. Now where we are some 20 years later, there's an extremely complex ecosystem of different types of institutions and actors. There's a really, really massive now set of industrial synthetic biology entities, companies, SynBio Beta, which is a huge industry-wide conference, which has been growing twice a year. Oh, and John Cumbers actually was also a LEAP fellow, going back to the fellowship program that Megan helped to create with Drew many years ago. EBRC, we've mentioned, is a big kind of research consortium of synthetic biology, but then also learning environments, right? Like iGEM, like How to Grow. Again, this class itself has been growing exponentially in recent years. And so these different types of institutions are really, really important as we think about what the future governance and cultures that are connected to synthetic biology are going to look like. And so how do we grow governance? All right. And so this is a kind of an interesting point when we talk about governance generally. Governance is not only done by governments. Actually, governance is something that happens at basically every scale of human organization, right? It happens in your family. It happens in classrooms. It happens within your lab. It happens at MIT. My colleague Marshall Gans at the Harvard Kennedy School, for those of you at HKF, he likes to talk about how structures are either explicit or implicit, right? Whether you know it or not, you're always part of some type of existing governance structure. Obviously, here in the United States, we've got a representative democracy. And also, interestingly, right now, like those types of norms are being challenged, literally as we speak. So governance is around us, often invisible, but sometimes visible. And there's different types of emerging systems of governance. I don't know if anybody here has any idea what this image is. Anybody know what this picture is? This little square diagram in the room.
Internet.
Internet, yes, thank you. This is the first kind of early architecture of the internet. And the point of this set of slides, that from that early internet architecture emerged some really complex systems of governance that attached to early computation networks. And those systems have grown and scaled and now are just a part incorporated into our daily lives. So we can see this kind of role and this impact that technical governance ultimately impacts cultural and societal governance and vice versa. And so how do we set these norms, right? Megan loves this XKCD kind of image showing all of these different actors in social media. What does the XKCD map of synthetic biology look like? We mentioned some of the entities here. There's industry folks, there's academic labs, there's How to Grow, there's iGEM. All of us are part of setting these different types of norms. And iGEM is a really cool story. And again, it has roots at MIT, has scaled globally. I think for many of the students that are in the global classroom, many of you are likely connected to iGEM. One of the, I think, most important parts about iGEM, this was not just a technical competition. It was ultimately a competition that also focused on governance and norms and cultural ideas and how do we actually do synthetic biology safely and responsibly. And there were rewards given for this. So this is one of the big kind of incentive structures that was built into iGEM from the very start. Yes, on the one hand, we want lots of cool biological parts, but we also want to award teams for thinking about ethical and responsible use of synthetic biology. So this, I think, was a big part of shaping. Thousands and thousands of students have gone through iGEM over the years, and many of those have launched their own labs, started their own companies. And so just being a part of a cultural paradigm, we have to think about practices and think about ethics and responsibility, I think is one of the huge successes of iGEM. And it's something, again, we hope is a big part of your experience and How to Grow as well. And so, this shaping global norms, again, really critically important. Megan herself was the director of human practices for many years as part of iGEM. And there are all of these different resources that have been a part of both iGEM, but also kind of global practices. This is a really great slide. I don't know how many years ago, George, this is of you, but this is not George lecturing to iGEM students about ethics. It's actually iGEM students showing George their ethical frameworks, right? And this is, again, what you all are going to be doing as a part of your homework this week. So this shaping of global norms, this has also been a part of the larger movement around community biology. Again, we talked about computer clubhouses, fab labs, there's also community biology labs. And many of the nodes that we talked about that are part of this year's class are in community biology labs that are from around the world. The Global Community Bio Summit, which is an event that I've helped to run and founded many years ago now. We've done 6 of these bio summits. Each year at the Bio Summit, we've done a large-scale global exercise where we've had essentially a question around governance, a large-scale exercise asking, what is our shared purpose? What are our values? What are some of our ethical principles? How might we as a global network govern ourselves? So all of these types of exercises, much like the iGEM global norms work, is all about shaping our larger culture and also our larger governance framework as we work together and develop these technologies. So let's now connect into our homework. All right. So we talked hopefully about, and hopefully I've convinced you that it is important for us to be thinking about ethics, practices, how to do synthetic biology safely and responsibly. And we're going to ground this now into an example. And this is a big part of your homework for the week. So this is a really neat case study, and it's linked from the website on this case of synthetic genomics. So as a part of this case study, they came up with a kind of a method, a set of methods that you'll be using for your homework. All right. So there was an overall kind of technical goal around synthetic genomics and synthetic genomes. But then there was this kind of question of, all right, what are different ways we can think about governance as it relates to this larger technical goal? Okay, so what's shown here is, what are these different kind of options for governance, right? And these can be connected to different types of goals that we could care about. So this now, I want to encourage you all as you think about your own project that you might be interested in. Again, it could be a computational project. It could be an AI and liquid handling robots project. It could be about design and textiles. It could be about education, something you care about in synthetic biology. There could be a principle that's involved. There's some that are shown here. So like one could be around fostering lab safety, right? So imagine you're doing a project around, soil microbes. How might you think about lab safety as a guiding principle, okay? And then there's many options for governance around that principle. So if you're doing like, soil microbes, for example, and you want to think about protecting the environment, there's a lot of different options for how you might do that. And there are a lot of actors that might connect to that particular approach. So going back to this synthetic genomics example, there are a lot of actors, right? There's folks that are doing gene synthesis work. There's users, organizations. All of these are different actors, and there are many types of actions that are possible. And so the what's shown here, and this is again what you all will be doing for your homework, for these different actors, like how effective might each one of these governance approaches be? So just zooming in a little bit as an example, it's like, okay, let's say we want to enhance biosecurity, right, and prevent future incidents. So this might be one of our larger principles is around enhancing biosecurity. So preventing future incidents, I'm just picking one at random. Firms must store information about orders. Okay, so this is like an example of of a type of method, and then each one of these methods was scored according to this little rubric. Okay, and so one of the kind of big points here is that there's no silver bullet, right? For any of the projects that you pick and can imagine, for any of these principles, there are so many different governance approaches and strategies you could take, and there's not one single thing that's going to be 100% effective. And so one of the kind of larger points is that there's always going to be some kind of patchwork of governance, right? It's not just going to be one thing. Through a lot of different approaches, hopefully you collectively have enough kind of governance approaches out there to have an overall kind of impact. And so, again, there's governance, there's kind of approaches around norms, agreements, like we talked about, banning bioweapons. There's legal approaches, and so many different types of governance approaches that you can imagine, from policy to community-based approaches and so on. And so, when we talk about your role, and again, as somebody that hopefully will, over the course of the next 14, 15 weeks, develop a lot of skills, develop a lot of capabilities, what we hope for you all is that, to remember that you're not just a technical person designing something cool, some new cool technology, you're also impacting the larger culture and set of norms related to the technology. And that there's a responsibility that you all are taking as somebody that is learning how to grow almost anything. And so, Megan's got a couple of kind of principles, which we really like. So don't do it alone, do it together. Not do it yourself biology, but do it together biology is something that we really advocate, which is also why, for our global students, you'll be working together with a cohort, with a node, with a set of instructors. And obviously here at MIT, we've got a cultural gravity here. Respect yourself and others, take responsibility, act responsibly. And then if all else fails, use common sense and ask questions. So you've got a lot of resources here that are a part of this course. All of our faculty, we've got 80 TAs, a lot of great folks that are alumni, instructors, and so on, that you can lean on over the course of this next 15 weeks or so. There's also some resources, responsible and ethical conduct at MIT. There's actually some really great documents here and beyond. So with that, I'm going to dive into the homework, and then we'll pause for some questions. So as I mentioned, right, this kind of synthetic genomics example was a good framework. And to Megan's point, it's also been adopted by others. The WHO has also used this kind of framework. So it's something that is quite effective at helping us kind of brainstorm and think about different governance practices. So, and TAs, if we can help mute whoever's speaking, that'd be great. All right, so your homework for this week, okay, and again, this is a requirement for all of you that want to be considered for one of our lab slots and also for our committed listeners, is to design a governance strategy to ensure an ethical future for a biotechnology of your choice, okay? So, and again, this is all going to be linked onto the class website as well. So again, if you go to 2025.hdga.org, the first class has all the information on there. All of this information is reproduced there. So, so step one is describe a biological engineering application or tool that you want to develop and why. So this is where, and this is potentially a seed for your final project. Again, it could be textiles, materials, computational stuff, biosecurity, biocontainment, AI and protein design, pick an area that you think might be exciting or interesting to you to start thinking about, both for your final project and also for this homework, okay? Next, describe a governance policy or goal related to an application or tool that contributes to an ethical future. Okay, so I'll give you an example in a moment. You know, connected to, you know, again, we talked about this idea of soil microbes. There could be a principle around how do I ensure that my engineered soil microbe doesn't spread, for example, right? How would I do biocontainment well? And then you propose a few different set of governance actions that could be connected to biocontainment of your soil microbes. And then you can score them. You can say, okay, for each of these different options, how effective do I think this is going to be? And we'll give you one example of a homework from a previous year's class and a student. Okay, so this was, again, about soil microbes. So this is a great example. And there were 3 principles that this student explored. One around biosecurity, right? So how can we ensure that the soil microbes you know, don't have a security harm? Another was around equity. If there are going to be benefits to these engineered microbes for soil, you know, how do we ensure that many people will benefit and it's not going to have an asymmetric set of impacts there? And then another one's around environmental concerns. So this student had 3 different principles, and then for each one of the principles had a different approach. So like, let's say for biosecurity, let's say we want to do monitoring, right? We looked then at a different set of actors. So the student, she looked at, you know, researchers, manufacturers, industry folks, organizations, and then for each one of these approaches had a little score, right? It said, is this going to be, you know, 1 to 3, most effective, moderately effective, or minimally effective, and then scored them. Okay. So this is an example of an output we want to see from you for your homework number 1. Okay. This is another example from another one of our students, Mickey. So this is you know, his governance framework around environmental protections. This is a different type of visual representation than was shown here, but a similar type of idea where you know, different types of goals. So one was promote equity and social justice. That was one goal. Protecting flora and fauna and the environment was another goal. Protecting cultural environments was another goal. So again, you can see this is sort of the approach here. What are your goals that connect to whatever your application is? And then what are the different approaches and how would you then score them? Okay. So this is basically an example of the homework that you all will be doing. Now, tomorrow in recitation, Lenny is going to go into a much deeper dive into all of this. And we'll go over his example from last year's class. You'll have a chance to ask questions as well. But that is your homework, your design homework that's a part of this week's class, okay? And you're also going to create your own student webpage and then upload this homework onto that webpage, all right? So that will be a part of what you all need to do to be considered for one of the slots for next week, all right? And then just to wrap up, you know, this is our homework from week 1, but we really want you to be thinking about these principles and practices every single week, right? Every week, you're going to be learning about a whole bunch of new interesting technologies. And as a part of your weekly documentation for your own student portfolio, we'd love for you to reflect as part of a little principles and practices section for each week's homework to think about, again, how might we think about different governance approaches connected to whatever technology you're learning about that week. And then for your final projects, you all will be contributing a governance exercise, just like you did for this week, but connected to your final projects. Okay, so that is what I have to share. I hope Megan is proud of me for getting through this material in hopefully a coherent way. And I will pause now for questions, comments, thoughts. That all makes sense. If you all were grokking that, can you at least raise your hands if you felt that was understandable? Okay, that's good. We love that. Any questions, comments about anything you all heard? Yeah, Cyrus.
For the websites, are you using things that are like automate to the GitHub structure, or is it just like to?
Yes. For how to grow, you'll be using Notion. So it'll be a little bit easier than GitHub. And again, tomorrow in recitation, Subin's going to do a live demonstration of how to set up your own Notion page. And there's a Google form. Actually, why don't I show this on the website right now? Actually,
we'll walk through everything step by step tomorrow in recitation.
So yeah, but let me just share screen. So if you all go to 20, so the URL is 2025.htgaa.org. Okay, so this is the 2025 website. If you go to this, it'll redirect you into the Notion page, okay? And so if you go into the class content, so all of the bootcamp materials, again, if you've never been in a wet lab before, we had a full week bootcamp in January. All of the materials are here. It's really wonderful. So you can see the detailed agenda, all of the slides are here, the resources, links to the recordings, all of it, again, shout outs to all the TAs. Again, a massive effort involved, and 9 hours of instruction. All of it is cataloged for you here. So if you're new to bio, please go and look through these materials. And then this week is our principles and practices. If you click into this right here, everything that we talked about from the homework is all linked on the website. All right, so the documentation is here. If you've got questions, these are your two go-to email lists for MIT and Harvard, HTGA 2025-TAs. That will go to our group of 16 TAs for our potential committed listeners. This email goes to all of our global TAs, and your assignment is shown here again. This is your practices homework. It's all linked here. Here's the example and so on. And then before next class, your personal Notion page. After you set that up, there's a Google form that's linked here. So you're going to put your name. You're going to link to your website, and then this is how you submit your website. This goes for our in-person students and our Harvard and our global students as well. And then in particular for the Harvard and MIT students, please do your lab training. So we're going to do a lab specific training tomorrow. You're going to go to the wet lab and get that training done. And then there's a bunch of digital training you have to do. And if you complete all of that, come back to the start of class next week, and then we will see how many of you are there at that point, and we'll kind of make a determination about the final class cohort. Okay, so that is the detail. And you can also have a little sneak peek about some of this content's going to be juggled a little bit. There's some moving parts here, but you can get a strong sense at least of a lot of the materials from the course website. It's again, really, really huge shout outs to Suvin for all of your leadership and helping to pull the web page together along with all the other TAs. So that is that. Any questions about logistics? No, there's been a lot today, everybody. It's been 2 hours and 40 minutes of intense synbio and logistics. So appreciate all of your patience. Any logistical questions? So one very important thing, did everybody who wants to be considered for a slot sign this form? This is the attendance sheet. So did anybody not sign this? Okay, that's this guy. Okay. So global students, please hang on. We're gonna do a little bit of a Q&A about any logistics in your questions. But to wrap up, and I know again we've had a very long day, so we can keep this part short. But if George is still there, George.
I'm still here, David.
Amazing. All right, let me add you in the spotlight. All right, George. Yay! Thank you so much, George, for also sticking with us, and Joe, I'm here as well. Joe, actually, I don't know if you want to come over. So for just the last few minutes, we thought we would just do a little bit of a panel discussion / Q&A with all of you. So I've got a few questions, but again, we really would love for our students here in person and also around the world to feel free to ask questions to George or Joe or myself or any of the TAs. So just to give you a sense of some of the questions that I had prepared, things like, what does synthetic biology mean to you? What are some of the things you're most excited about for the future? We can start there if nobody has any questions, but I'll first pause for questions in class. Any of the students here have questions about anything? This is an AMA, ask us anything. This is a very prized moment. You've got George Church's undivided attention for the next 20 minutes. What do you want to ask George or myself or Joe? Any questions so far?
I see a bunch of hands up.
Yeah, why don't we go globally? Hadi, do you want to unmute? You're first there. Yeah, go ahead, Hadi.
Hello.
Yeah, ask your question.
Can you hear me?
Yes, we can hear you.
Yeah, first of all, I would like to thank you for MIT to, celebrate. I'm so nervous. Yeah, I would like to thank you for the efforts and all the preparations that were made for this. And I've joined since 2 a.m. in the morning, so I woke up early to be here, guys.
Oh, thank you.
And that's for also. And I would like to be a committed listener. Yeah. So my question is there currently in the world right now, are there any, some sort of seminar or maybe some sort of gatherings all around the world to discuss this biotechnology advancement, like every year or maybe every 3 years or something. And if there is so, do they also, incorporate all of the countries in the world, like China, Russia, even Palestine also maybe? I'm just curious.
Yeah, so a great question. There's definitely a lot of different types of global gatherings at different levels, depending on the communities involved. There's big academic conferences. For example, SEED is a really good synthetic biology conference that has a lot of academic research labs that have been participating for many, many years. There used to be a conference called SB, Synthetic Biology 1.0, that went all the way up to 7.0. The BioBricks Foundation was behind organizing that. And actually, this is, again, connected to Drew. The Asilomar meeting that I mentioned is another global event. I don't know, George or Joe, if there are other conferences that you both want to mention.
Well, you've mentioned already iGEM and SynBio Beta, which meet once a year or twice a year. There's some less regular ones that come up as needed. Yeah, there's no shortage of meetings, I would say.
Yeah. Also to mention too, I mentioned briefly the Bio Summit, but we've hosted that at MIT 6 years. We're going to be doing a 7th one. That is a really great space for a lot of the folks that are doing more kind of grassroots biology. But George,
I think part of the question was whether it was equitably distributed, and it's not quite because of the cost of travel and so forth. But I think some of them are hybrid meetings. I think that helps make it a little more equitable.
George, just to kind of get your thoughts on this, and also same thing for Joe. What are both of you most excited about right now in the field? I think as we've been teaching this class every year, I think the machine learning, artificial intelligence side and the protein design has really exploded the past number of years. Joe mentioned a couple of topics at the start, but George, for you, what are you most excited about, both right now and in the near-term future for SynBio?
Yeah, well, the main one probably is the impact of AI on protein design, and that's the main. I think that's the best application of AI, period, so far. But it's spreading to adjacent things like RNA and vaccines and cellular differentiation and so forth. And then I'm excited about things having to do with those complex developmental biology, not just curing aging and making bringing back old DNA, but the idea that we could get developmental biology to be a programming language. So that Joe showed an example in his intro of how you can model developmental biology with things like game of life, sort of cellular automata, but how about actually making things from scratch? We're making organs now, but not from scratch. We're making them by engineering a pig that's more compatible with humans. But what if we could do it from scratch? That would rival 3D printing in a big way. Instead of having one print head in Cartesian coordinates, you'd have trillions of print heads per cubic millimeter. Anyway, so that's something I'm excited about.
Yeah, and just to say too, Ron Weiss, who is going to be helping to lead the genetic circuits part, has also been doing a lot of great work in organoids, and we'll speak to some of that in the genetic circuits class. But yeah, Joe, I know you already addressed this a little bit at the start, but I don't know if you've got any build on any of George's comments.
Question?
Oh,
yes.
Yeah.
Now go. What is currently the biggest roadblock?
Great. Yeah, great question. So the question was, what's the biggest roadblock to the field? I don't know, Joe, if you want to.
Yeah, I'll maybe tie both of those together. So I agree with George. I think protein design is just so transformational. What's happened over the last couple of years, and I think we're all eager to see how excellent that can get, how accurate that can get. I think I'd say one of the roadblocks in protein design right now, from my perspective, at least some of the things that we work on, is that AI is trained on data sets that are basically originate from crystal structure, meaning from a frozen state of a protein. Proteins move a lot. We have large classes of proteins which are disordered, meaning much of them are moving all the time. And so I think one of the next frontiers is molecular dynamics, AIs that can understand the dynamics. Those are very large compute problems right now. Just to do an MD run on a single protein ligand pair can be something like $100 of compute or more at scale. And you want to do like millions of those. So that's a barrier. I think another area that both George and I have been interested in a long time is DNA synthesis. That's progressed to a point, but we still can't make entire genomes at any kind of reasonable cost. We can't make entire genomes at all, above let's say a couple of megabases or so, and we can't make long constructs. If we could, we could start to make things like systems of enzymes, things like polyketide synthases, basically nature's chemical factories for making all kinds of very difficult to synthesize molecules we can't synthesize as organic chemists. And so I think the potential to transform chemistry is also super exciting. So those two, yeah.
That's great. Yeah. Yeah. Thank you, Joe. That's awesome.
Oh,
Yeah.
sorry.
Go ahead.
Kind of a follow-up, I guess, all three of you. You mentioned it in a seminar 50 years ago, yourself, 50 years, but they stay the same problems being discussed.
Yeah.
How did your expectations when you started in the field match up to reality? What surprised you? What has failed to happen?
So the question is, kind of how has, I guess, how have our expectations in terms of like what we thought about the field versus how it's evolved?
Yeah.
George, I don't know if you want to, how's the field been evolving, according to your expectations, George?
I think it's met expectations, maybe exceeded them. I mean, it's on an exponential. Just reading and writing DNA and everything you can do with that is on exponentials. But if you're expecting an exponential, then it met expectations. But most people weren't expecting it. They were surprised by it. I'm also pleased by, I don't know whether it's expected or not, by the fact that we've been much more proactive about safety, security, and ethics than, say, the Manhattan Project. I realize that's a very low bar, but I'm just pleased that or for that matter, robot wars, or I think that this embracing of human practices is not just specific to our course.
Really quickly to follow up on that, and both, George and Joe, both of you, you both have talked a lot about the impact of AI and protein design. I'm curious what your take is on like practices, governance. Like, are there things that we should be thinking about at the intersection of these two technologies, AI and synbio?
Yeah, it's very important. In fact, David Baker and I wrote a little paper in Science to draw attention to this. I think there were two things that we proposed mainly. I mean, distilling it a lot. One is that the system that we set up for doing surveillance of DNA orders should be part of the protection on protein design. And the second thing is that these should all be recorded. I mean, I don't think these are completely brand new ideas. It's just that for the protein design community, they were important to point out. So if you record these things, and even if you can't figure out whether they're threatening or not at the moment, if something later happens, you can go back and track it. And I think that was actually on one of your slides towards the homework end of things, something along those lines. But anyway, that's what David and I said.
George, if you don't mind sharing a link to that paper into the chat, that'd be awesome.
Yeah, sure.
And
Yeah, I'll do that.
if that's not already on the resources part of the course webpage, it'd be great to add that there. So that'd be an awesome great paper. Yeah. Yes. Yes. Yeah. 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 So the question is, George, and I think Joe for both of you, but the question is about genome write versus read, and kind of where we've been with the last 10 years of genome write. How's it been going, and where is it heading? George, I don't know if you want to. You've been
Joe and I.
Yeah.
Joe and I talk about this a lot. We're probably pretty aligned. So I think they've both undergone roughly a 20 million fold improvement in cost effectiveness and quality. Although I would say that but they're different categories. So we can now get a perfect human genome, telomere to telomere. There's only 2 or 3 of them have been done, but we've done it. 10 to the minus 10 error rate. We have not anywhere close to being able to synthesize that, but nevertheless, it's improved tremendously. I'll let Joe finish the sentence there.
Building is harder than writing is harder than reading. And I think that the reason is that reading is you can just in parallel you can read all different little snippets and then you can stitch that together. And some things in the world work that way and some things don't. And some things we don't quite know yet. So like back to molecular dynamics, if we take, thousands of 1 nanosecond snippets of molecular dynamics, we actually don't know yet whether we can stitch that all together into some long thing. But it does work that way in reading. In writing, it turns out that you can build lots of those little snippets, but then putting those all together accurately with no errors turns out to be difficult. So there's nothing in physics that says it's not possible. We have, in transistor world, we've pushed that technology, and the error rate on writing a transistor, meaning the error rate of building a transistor and working is on order 10 to the minus 12 or so. And so, there's nothing that we know of that prevents us from doing that, but we're not quite kind of there yet. Say that one more time. Correct. I mean, yes, right. You've got, at the end of the day, you've got to build a genome, at human size, you've got to put 1 billion things together and with no errors, essentially, or a very small number of errors.
So class 9 is our building genomes class. So I think again, we've got George, it's going to be epic. Yeah. John Glass and Jeff Boki are all here. And so that'll be, we will go in just gory detail into that in week 9. Yes. And again, go ahead.
On the business side of SynBio, with EcoBio, we're just getting like a massive valuation and
man,
struggling to
roller coaster. Yeah.
And now with Colossal Biosciences having a massive raise with a humongous valuation, is that good for the general SynBio industry, or is it risky if you can't live up to those promises? How do you feel about the general industry?
So George, if you just for everybody, so the question is about synthetic biology and industry, reflecting on Ginkgo having this huge valuation, then the roller coaster they've been on, Colossal having a huge valuation, which George, you're intimately involved in. So the question is, how do we feel about this kind of roller coaster ride, and where's this all headed?
Well, I think we need to include in the SynBio community some on the edge, which is the biotech companies that kind of preceded SynBio and have taken on some of it, but not the core of it. I mean, Colossal isn't yet going down on its roller coaster, but so that's good. Ginkgo will hopefully rise to the stratosphere again. I think we should. I always hope that industries avoid bubbles. It's actually fairly disruptive to have a bubble. We should not encourage it. Because people get discouraged when it pops. I think steady growth is a much better model, but I don't know how to do that because we have this exponential, we have all this enthusiasm. People are going to discover new things like the intersection of AI and proteins and et cetera, and there's going to be enthusiasm, and it's really hard to curb, as they say.
Yeah, but I think in biotech this is not unusual. You know, 40 years after, George will tell me the right number, but you know, Genentech, back in that era, you know, there were 20 companies like that, and the first ability to splice DNA felt revolutionary, and people thought you could do everything with that, and it turned out that you could only do, you know, a few things with that, and you know, 1 or 2 companies out of 20 survived, and then you have another cycle and another invention, and then you have a boom again because you think you've got most of what you need. And I think you know, probably ginkgo was somewhat like that, which was, it was probably, the thought was we have enough if we can just recombine existing parts from biology. The truth is it's probably was not enough, and we really needed what we have now. We're starting to have the ability to design new enzymes, to design new parts that didn't exist. So I suspect there's going to be, and there are already signs of another boom in this area, and that's the nature of biotech, I think, which is there's a lot of layers of the onion. We don't know all those layers. My wife has a theory that it's infinite, and I told her it can't possibly be infinite, but it does seem that way, that we always are discovering something like very fundamental. But that, I think it's embedded in the very fabric of biotech.
So we are at time. I apologize for all of, oh, George, go ahead.
No, I was just going to say that this is going so well and we are at time. I would love to do this almost every time. Or maybe we can set up a special time. Okay?
I know.
There's at least 12 hands raised. So
I know. So for listeners, I'm going to hang and so please stay for the listeners. For the MIT Harvard students, again, thank you so much. I know today was a marathon. Appreciate all of you. If you've got any questions, I'm not sure where Subin is. I think Subin's outside, but feel free to hang. Ask me any questions as well. I'll stay if you have any. But please come to recitation tomorrow, 5 o'clock in 359. And just one more big round of applause and thanks to all of our speakers and all of you all today. Thank you guys so much. And we hope to see you tomorrow for our next step on this adventure. So, all right.
Awesome.
Jay, thanks so much.
Thank you. Awesome.
All right. So everybody globally, just hang tight for 1 minute. Okay. Or like 2 minutes. Keep your hand raised. I'm gonna stop this recording, but I'm gonna stay and answer questions. So please hang on. So the reason that we have this organized by nodes this year, and again, it's you know, because we're still kind of building the airplane as we're flying it, as I was describing earlier, we have 8 official nodes this year. And unfortunately, for example, we don't have one in Africa, right? But we do have one in Latin America, but it's only in Ecuador, right? So it's not a lot of geographic diversity, but there is like time zone diversity. So so one of our goals this year, and we're doing this for the first time, for all of you that are committed listeners, we want you to join one of these virtual nodes, and then you'll learn alongside a cohort of virtual students. And so again, you know, this is sort of just where we are in, you know, kind of building this airplane that we're flying. We would love for all of you to be able to go in person someplace to do this work, but that's just realistically not the case this year. There's only 8 physical sites. So if you happen to be close to one, wonderful. There's, again, TAs there that can help support, and you know, you'll have a great experience there. But unfortunately, if you're not geographically near one of those locations, join a node virtually, and then you'll be able to learn and at a minimum do the virtual homeworks. So again, all of the protein design work you can do virtually, all of the work programming robots you can do virtually. And, again, Adrian, a number of others have been really kind of pushing the frugal science work. So there may be some ways for you to do some of those experiments regionally, locally to you. And we'll all kind of work and figure out that together as we go. And so this, again, the be flexible, be patient for, the global students. Our emphasis, my emphasis is first and foremost for the MIT Harvard students, but we really, really want to support as much learning as we can for the global students. So right now, the design work, the coding work, the robot work, all of that can be done remotely wherever you are, as long as you have a computer and an internet connection. But the in-person work, unfortunately, is going to be more limited to folks that happen to be near one of the physical nodes. So that's kind of just where we are at the moment. But Richard, thank you so much. Next question, Mikhail.
Hello, hi David. Thank you very much for the lecture today. It was absolutely amazing, 3 amazing hours. So my question is kind of I would not say practical, but more theoretical one. So how do you think will in silico either experiments or clinical trials or stimulations like in total in the future, would they substitute the ones that we have already? So maybe the organs on a chip, the basically the normal stuff that we have right now.
Yeah, that's an awesome question. So, there's kind of this pipeline, right, of like in silico, in vitro, in vivo, right? In silico, pure simulation. In vitro, things like microfluidics, organ on a chip. I've done a lot of that in my own technical career with artificial guts, artificial noses, things like that. And then in vivo, doing actual experiments, in animals, in living systems. And so it's a really great question. I think there's a lot of interesting simulation work that's happening at the cellular level. So, for example, in the Cell-Free Systems Week, Kate Adamala, she'll be talking about what are called synthetic minimal cells. And there's a whole big global program around what's called Build a Cell. And they're also doing a lot of simulations around cells. So that's some area where I'd encourage you to kind of look. But the modeling is really hard. Like we don't have like a really good, in the protein design fundamentals class, we'll learn a little bit about this, but, we don't have a good molecular representation of water, right? So it's like, a lot of the modeling is actually really, really complicated. I think though, with a lot of the AI tools that are being developed, a lot of that can be accelerated significantly. It's a really, really great question that I think, we can explore throughout the semester. But I'll point you initially to Build a Cell as one place where I know they're doing some really interesting simulation work. But thank you for the question.
Okay, thank you very much. Thank you very much.
Yeah,
Hey,
thank you so much.
David. Sorry,
Yes.
Dominika here. Before you move on to the next question, can I perhaps introduce GenSpace a little bit? Because I think we might
Yeah, of
have.
course. Please go for it.
Okay, great. Do we not have a slide or?
I covered the slide in my lecture, but I can open it again.
Okay, no worries. Okay.
Yeah.
Okay,
So
I just want to bring a little bit attention to it. Just
yeah,
like
of
together.
course. So
all
let me
the.
share this page. So we talked a little bit about the nodes. Yeah, nobody spoke up. So I kind of talked briefly through it, but yeah, go ahead, Dominika. You can speak to
No,
Gen.
okay, cool. So, GenSpace is based in New York, founded in 2009, the world's first community biology lab. We will be meeting weekly in person on Tuesdays, 6:30 to 8:30, to do the lab work together and homework reviews. So join us by applying as a community project member on the website genspace.org. Genspace.org. That's it. That's my announcement.
Hey,
Thank you very
perfect. We
much.
love that. Thank you so much. All right. William, go ahead. William Elliott, are you there?
Hi, sorry. Yeah, thanks again for today. Mine is just a much more like practical question, but in terms of the homework and the recitations, I'm not really familiar with the format on that. Is it due tomorrow or is it due next week? And is it kind of a verbal session or is it kind of virtually submitted?
Thank you for the question. So, okay, so basically for the listeners, this is how this is all going to work, okay? Next week, after you do your homework, okay, for those that, complete the webpage, do the homework, and so on, we're going to look and see at the total number of listeners that we have, and then we're going to have you all pick a node to be associated with. So again, there are 8 nodes representing a good diversity of time zones around the world. And so based on the node that you pick, that node will have its own recitations for that node. So for example, and these are actually shown on the website. So as an example, let me give you let me just share my screen again and give an example. Okay, so if you go to the course webpage, here are some global nodes. Great. So it's like here's BioClub Tokyo in Japan. Class reviews Thursdays, 2100 JST, homework reviews 2100 JST, etc. Right. So each of the labs has got their homework schedule time. So you can either pick a node that's in your time zone, or pick a node that has the recitation time that works for you. Okay. And basically, we recognize that this 2 to 5 Eastern time is terrible for Asia, right? It's like you guys are waking up in the middle of the night. It's really, really hard. But basically, we're going to post the recordings as soon as we can when the class is finished. And the attendance for you to be considered for a certificate, your attendance is not going to be necessarily about your participation in the global lecture, although we would love for you to join if you can. It'll be about your participation in the node, okay? So let's say you're in Asia and you're part of either the Korean node or the node in Japan. Those weekly recitations, that's where you're going to be evaluated and where you should be showing up, okay? And so the homework that needs to get done, you'll work on that together with your node weekly, all right? Now, the MIT recitation that happens, in general, if you're able to join, that will be most ideal, because we do a lot of instruction about the homework that's happening that week. Again, a lot of the lab work that may not be as relevant, but some of the recitation instruction will be. Again, all of that will get recorded and put onto the website, as soon as the class is over. But the most important thing for you once you become a committed listener is to be really diligent about joining the sessions that connect to your node. And again, we understand totally, especially if you're in Asia, these times are like in the middle of the night. Watch the recordings as soon as you can, and then participate with your cohort within your node.
Cool. Thank you.
Okay. Thank you, William. Eduardo, go ahead.
Unmute. Yeah, no, I guess my main question was on how should I say, it was since we've drawn throughout the class so many parallels between AI and SynBio, right? And they both, because obviously they do share a lot of things, like not only do they, as you were saying, like it's either, at least in its inception, it's either something that's going to solve all the problems in the world, or like going to cause like the, I don't know, doomsday for some sort of doomsday scenario, right? So, sorry, can is my
Yeah, what's your question? Yeah, we can hear you. What's your question?
No, like, exactly. The question is like, how do you think we can reach better to like how can we communicate better to like the public, to like not have to like sort of like find a middle ground between like panic and like overblown excitement,
Yeah, it's a great question, and again, I think it's I would encourage you to think about as a part of your homework, right? It's like a perfect question for you to consider as a part of your homework, right? And I think the answer is sort of like what I was sharing in the presentation. It's going to be a patchwork set of solutions, right? There's going to be cultural norms, there's going to be technical ways, there's going to be agreements that occur amongst research institutions. There's going to be policy recommendations, right? There's going to be multiple levels at which the work needs to happen. And so again, I think, again, not to be too, what's the word, pedagogical, but I think, that's something that'd be real. It's a good thing for you also to think about in the context of your homework. I would really think about, what are, if you come from a background that's more technical, you could think about technical solutions. But if you come from a cultural background, there's a huge role for art and design, right? Like, how can artists help communicate about different types of biological futures, right? How can we ensure equity if you want to come from a community organizer perspective, right? So all of those, I think, are various modes that are important.
Okay, thank you.
Yeah, thank you for the question. Helen from New York. Helen, NYC, are you there? Okay, we'll come back to Helen. Gustavo, how about you, Gustavo? No, Gustavo. Avantika.
Sorry. Got it.
Oh, sorry. Is that Gustavo?
Yeah, sorry, it took me a bit.
Okay, go ahead. What's your question?
Hi, so I had a question regarding the final project. Are there any limitations to it, or because I'm already more involved in terms of genetic engineering, and I would like something that is maybe not directly with bacteria, something that is a higher level of engineering? I don't know if that's a possibility.
Yeah. So thank you for the question. So for final projects for global students, this is actually really important. Again, the majority of you, unfortunately, are not going to have access to a physical wet lab. And even for the physical wet labs for each node, each node is going to have different capabilities, different access to resources. So it often is likely that you may have to be doing more of a design type of project. And you can still do a lot of work with design. You can still do a lot of work on DNA construct design, protein design, and so on. The way that we structure our final projects, we break it up into aims. So each student has to share 3 different research aims. One that is like kind of a fundamental basic one, and then a second and third aim that are increasingly more visionary. So I think if you've got experience in genetic engineering, we encourage you to have really visionary aims or really visionary project, and then some type of experimental goal that you feel like you can accomplish or test either computationally or in the lab. So we'll help you scope out those aims over the course of the final project process. But that's going to be really one of the big areas is, if you've got access to a lab, scoping something that you can accomplish, some set of experiments that you can accomplish in the lab. If you don't have access to the lab, then certain things you can design or explore computationally. Those things we really encourage. And again, if you look at final projects from previous years, there's some really amazing projects that students have done despite not having access to a wet lab. So thank you for your question. Okay, let's go to Avantika. Go ahead.
Hi, David. I have a super quick housekeeping question, but it's about if you are local to Boston or the Cambridge area, is your node then MIT or is your node GenSpace?
Great question. Yeah. Unfortunately, your node is GenSpace. Okay. So again, our priority is focusing on the MIT Harvard students at MIT. However, we'll say this. If you are, really excited, got a lot of passion, a lot of interest, hang with us. Just let's see how things evolve over the next couple of weeks. Again, it was really crazy. Like the response this year was really enormous. I mean, we had like 12.
Completely understand. Yeah.
Oh, no, but just to say, this always happens where there's a whittling process, right? Like we had 1,200 people apply. I mean, there were at least like 500-600 of you over the course of today, which was really amazing to see coming in out of the virtual classroom. But as things go, it's just things kind of whittle down, right? So, so for those of you that are in the Boston area, there will likely be space for you to join the class actually in person. So just kind of hang with us, virtually, keep joining the virtual classroom. And then, as the class goes forward over the next few weeks, we're going to see just a whittling down of students, both globally and locally here at MIT. And so it may be that there could be classroom space for you to come in person for lectures and recitations, and there may even be lab space. Okay, so it's something that, again, if you're passionate and you really care a lot and want to be kind of connected in particular to MIT Harvard, just hang with us and we'll kind of see how the situation evolves in the coming weeks. Okay, but for now, join the GenSpace node, work with the students at GenSpace, but just we'll see how things go over the next couple of weeks, and you very well may be able to, kind of sneak your way into working with us in person at MIT.
Thank you so much.
Great. Trevor Campbell.
Hey, David. Thanks for going through the nightmare. I know this is a logistics hell for you, so I appreciate that.
Oh, it's not. I don't even know how to describe it, but it's like, good problems. I mean, seeing like, the Zoom room, like potentially crashing, like it's like horrible, but also like amazing, it's like, so grateful to all of you for coming and being a part of this. So thank you for being here.
My question was the same as Avantika. I'm based here in the Boston area. Something to consider too, even for this or for the future. There's definitely partner organizations here in Boston that probably would be willing to extend wet lab space. Off my head, I'm thinking there's
Boss Lab?
definitely
Boss Lab.
what's that? I mean,
Boss Lab?
Hash Bio is one, but Mass Bio, their new bioversity, this is right up their alley. I'm sure they would be willing to extend some support for this, and I can help spur that I can help spur that conversation if that's something you want to have.
Yeah, I think, again, for the folks that are listeners in Boston, very, very happy to talk about that. Again, the 8 nodes that we have to start, the reason why it's those 8 is because there's a very experienced TA that's taken the course and is ready and available to help and support and instruct. We want to expand and have more nodes for sure, like just absolutely for sure we do. And so it actually would be really, and there's also Boss Lab here in the greater Boston area. So there's a lot of labs and spaces that actually could support the growth. So let's be in touch. Again, I think there's a possibility for you all to join in person here at MIT, but I would love to support a non-MIT lab space. That would be really, really wonderful. We'd love to see that. So
let me know how I can help.
Yeah, and actually, again, as the course unfolds, we rely a lot on the global TAs for leadership. So if you're interested and want to help set something up, again, we'd be happy to kind of chat with you about that. Just pause really quickly for the global students. I just want to chat really quickly with some of the in-person TAs for just a minute. So just
thanks.
Okay. All right, everyone, I'm back. So sorry about that. Who was up next asking a question? And Kana, thank you for being patient. We're gonna just get through this. Engin, up next. Engin, you have a question.
Hi. I have two questions. One of them is to be dedicated listener, a committed listener, sorry.
Yes.
The first one was like you need to sign up form for the Google form or something like that. So to apply this course, we already, I think.
Yes.
So, so was there some, because I checked, but there was no section like, do you want to be a committed listener or something like that? So what is the process? So, like we need to do homework. So, and we need to go to lab or something like that. So after that, the process is automatic or we need to do additional?
Yeah, so basically the process is do the homework. That's the most critical thing, okay? Make sure you have a student webpage, make sure you've got your your ethics homework done. Make sure you do the homework properly, okay? And then and it's due at the start of class on Tuesday, okay? On Tuesday, we're going to look and see everybody that did the homework, and we're going to see what that number is. It could be, 500 or it could be 50. Okay, we have no idea, right? So based on the number of students that actually go and complete the homework and produce a student webpage, the TAs, we're going to have a meeting and we're going to discuss what we're going to do, right? If it's like an overwhelming number, we may figure out a way to either screen it or have it be smaller. If it's a reasonable number that's divisible by 8, I mean, I think, we're going to be able to take hundreds of students. I mean, again, if imagine even if we have like, 300 students, let's say, we can have like 30 students per node. That's not like a crazy number because that number will also diminish. Again, despite all the intentions and excitement at this first stage, there's always kind of a decay function that occurs. So we're very likely, if you do the homework, you will then, after next week, then be assigned to a node. You will pick a node to then start showing up and doing your homework with for that week and every week going forward.
So we need to choose the node before we finish the homework?
No. You're not, no node choosing is happening until after you do your homework. So all you have to do for now, if you want to be a committed listener, is do the homework, and then we will then immediately get back to you and initiate a process for the students to choose nodes, depending on how many students we get, right? It could be, if it's an overwhelming number, we may have to do another screening process. We don't know. So this is a part, again, where we appreciate your kind of flexibility with us as we try to accommodate as many students as we can globally.
I see. The second one is the Notion website is a must, or if we have already
must. It's part of your homework. If you do not do a Notion webpage, you will not be considered for a committed listener slot. That's part of your homework.
I'm asking, like, if we already have the personal website, we can't.
No, it's not about your personal website. It's about making a web page for the class. This is your student portfolio for the class. Yeah.
And it should be in Notion. So I'm asking.
It has to be in Notion. Yes.
Okay. Thank you.
Great. Thank you so much. Nicole Fu, you are next.
Hi. So I have two questions. The first one is because I'm not from biology background. So my goal for this class is kind of like expand my interdisciplinary knowledge and learn from like the top researchers. So I'm just wondering, is there like any projects, books, or like research areas that you're like really exciting about different intersections of different areas that you recommend?
So I would recommend starting with the class webpage. So again, if you go to 2025.htga.org, if you go to this website, okay,
yeah,
which is a class website, if you open that up, and this is very important for those of you that have never done any wet lab work or are new to biology, there's two big pages here: Bio Bootcamp 1, 2, and 3. If you click into these, there's all of this critical background for the class. 9 hours of lectures, papers, all kinds of resources for you to look into just to get the fundamentals. And then Beyond that, you can look at the course webpage. Every single one of the classes has got all kinds of materials for you to look at and have a feel for. So I would just go and look at the website and see what gets you excited.
Oh, okay, I see. Thank you. And my second question is not like really academic focused. So because I'm kind of interested in like potential applying the MIT Media Lab in the future, so I'm just wondering, do you have like any kind of insights for what kind of applicants, like the traits of the applicants you are really looking for?
Yeah, I mean, I would say, be excellent, just work hard, be excellent, do great work, and all the doors will open for you in life. Being a part of this class is a great example and a great start. Work hard. This class is a great place where you can meet a lot of key instructors, faculty. In life, doors open when you go and you knock on the doors and you try to open them, and you go and live as enthusiastically as you can and with as much passion and initiative as you can. So you being here, you asking these questions is already a great start. So keep doing it. Keep doing what
Okay,
you're.
thank you.
Yeah.
Thank you.
Great. Thank you. Okay, Alexander.
Hello. Well, first of all, thank you for everything. I would appreciate all of your effort. I would like to ask, will there be any monetary cost for committed listeners to attend the NOTE or the NOTE web lab? And if so, when will we receive the information about it?
Yeah. So you will all know we're going to make decisions about nodes and allow you all to sign up next week. Okay. So, and if you're in Quito, which it looks like you are you connected with Benjamin?
No, not already. I don't know.
Oh, you are in the very lucky circumstance where you likely are going to be able to do in-person work with Benjamin. So, so reach out to them. Again, you can send an email to the larger TA list, which again is HTGAA 2025-global TAs at media. So this is the TA list. Feel free to send an email there. You happen to be like close to actually one of the nodes, so that's a great luck. And yeah, I think Benjamin will be very excited to connect with you.
Okay, thank you.
Great, Melanie, also from Quito.
Yeah, hi David, nice to meet you, and thanks to everyone from HTGAA for knowledge. I have too many questions, but they are related. So, okay. First, if due to the schedule reasons, because I'm working at the hours that you put for the recitations in Ornato and also for the lab class, it is difficult sometimes for me to attend to the recitation. In the case of it will be in the Noto. Can I join to the virtual ones? And our recitation is starting to be considered a commitment listener. And what will happen?
Sorry.
Okay.
Let me just answer that.
Okay,
question at a
one by
time.
one.
Let me answer the question first.
One by
Okay.
one.
So if you have a schedule conflict, and this goes for anybody, any potential committed listener, fear not. Okay. Ideally, we want you to be working with your node. So since you're in Quito, Melanie, if you're able to work with Benjamin, like that's the most ideal, because you can go in person, work in the lab. But if the timing of the Quito global recitation is a conflict, then find a node that has a time that you can join virtually. Okay, so I'm going to share the website again. If you go down here, you can see each one of the nodes shows their recitation times, their meeting times. Okay, so if you're not able to join the times that are shown for Quito, Ecuador, then pick one of the nodes that actually has a time where you're able to join. Okay, and then you can work virtually with that node.
But it will be virtual, right?
It'll be virtual, yes.
Okay, and related to the tomorrow recitation that I saw on the email, if we don't have a signal node yet, so it will be virtual as well.
If you can't go to the recitation, try your best, but if you can't, don't worry about it. Again, the attendance is only going to matter really matter once you start picking a node. Once you pick a node, that is where, if you want to get a certificate, showing up every single week is going to be very important, and the attendance will be tracked at your node, whatever that is. So if you can't make the recitation tomorrow, it's okay. We're going to be sending out the recordings later, just like we will for today's class.
Okay, and I don't want to be so negative, but in the case that due to the capacity, I'm not assigned to the node on Quito on the San Francisco University, can do the practical part in my laboratory if I have all the necessary material, because I'm working as a research technician in a laboratory, in a research laboratory. If I also have a OT, an Opetron OT too, I can use here, or I have to go to the San
That's
Francisco.
interesting. That's interesting. So I would say in your case, talk to Benjamin first. There may be some ways where you can collaborate with their node somehow. So again, this is sort of a, we were talking about this earlier with the students from Boston, right? I know there's excitement to set up more nodes, which is awesome, and we definitely want to do that. But we don't want to do that unless there's like an experienced instructor that can work there. Right. So if there is not one that's able to work specifically in the lab that you're talking about, then, it would not be an official activity, or it would not be part of like the committed listener approach for our class. So I would work with Benjamin though. If Benjamin is able to support you in your local lab, then, or if the TAs that are in Latin America can support that lab work somehow, then, we can potentially figure that out. So I would reach out to Benjamin first and chat with him.
Okay, I'm just asking because sometimes the schedule is difficult for me to move to different places.
Yes, yeah. Start with Benjamin and we'll go from there. But thanks for the question.
Okay, thanks.
Next question, Folesiwa. I apologize if you can, how should I pronounce your name?
Folashewa.
Folashewa. Okay.
Yeah. So just a question. At what point does precision therapeutics for neurological disorder intersect with synthetic biology?
Say it one more time. At what point is what?
At what point does precision therapeutics for neurological disorders intersect with synthetic biology?
Got it. Great question. It intersects a lot. So, just as an example, here at the Media Lab and at MIT, one of our faculty is Ed Boyden, who leads the synthetic neurobiology group. Also, Deblina Sarkar, who leads the oh my God, I can't pronounce it. Anyway, her group title is very complicated, but it also focuses on brain and neurons and synthetic biology related to the brain. And so, precision medicine is a huge part of the promise of the future of synthetic biology. The idea that we can, do real-time or precision reading and diagnostics, and then being able to develop different types of precise and custom treatments. That's a big part of the whole promise of the field of synthetic biology. And specifically as it relates to the brain, there's a lot of really exciting research that's happening. And again, just those two faculty, I'll put them in the chat. Ed Boyden is one, and Deblina Sarkar is another one. They're both doing really exciting work, I think, at that interface of synthetic biology, the brain, and also precision medicine. Thank you for the question. Okay, let's go to Ansh. Your question.
Am I audible?
Hello. Yes.
I'm not sure if I'm audible. My question is related to gene therapies. So like gene therapies have shown promise in treating multiple age-related diseases, such as in mice, the heart failure, kidney failure, and diabetes, and sometimes obesity. So what do you see as the primary challenges in translating these therapies to human patients, and how we might overcome them?
I'm sorry, I couldn't hear the last part. So which two fields? Say that again.
I'm just saying that, what do you see as a primary challenge in translating these therapies to human patients, and how we might overcome them? Yeah, that's my question.
Got it. Gene therapies. Yeah. Great question. I think there are a lot of
in age-related diseases, right? So yeah.
Gene therapies and what?
In age-related diseases.
Age-related diseases. Okay. Yeah. Great questions. So there's of course technical challenges, right? Any kind of gene therapy, there's always questions around delivery, targeting, specificity. There's always questions around good models. Like, do we actually have like good longevity models for us to actually test and for us to develop gene therapies to interrogate? I actually personally am not quite as up to date on the state of the latest kind of technical state as it relates to gene therapies and longevity, but some of the types of issues are around specificity, delivery mechanisms. Are there good models available? And then again, this goes back to the governance questions we were talking about. There are really big questions around regulations and regulatory domains depending on where you are in the world doing this work. So. I think there's a lot to consider in that particular area. And I would actually encourage you to look into that for your homework question. I think that's a great area. Synthetic biology and longevity, really, really interesting. You could come up with some really cool governance principles around that set of biotechnologies. That'd be really interesting. I'd encourage you to look into that.
Yeah, sure. Thank you very much for your time, and thank you for very much for letting me in the HTGA. So yeah, thank you.
Yeah, wonderful. Thank you for being here. Javier, you're next.
Everyone, thank you so much, David, for this amazing course. I have two questions. The first one is related with iGen. Actually, I am the current ambassador of iGen in Latin America. I am very happy to see a lot of Latin American fellows here in this course. I know that will be a lot of activity this year. Well, I want to ask you if there will be any possibility to maybe share the final projects within the iGen platforms. I can ask to the team of Paris, and we can try to help to the HTGA course maybe.
Yeah, I love that. So again, Javier, reach out to Ben. Again, I think that's going to be our main Latin American node. I think you absolutely should share your work to any of the other kind of students that are part of the node. I think that'd be really exciting to share that work. And we can definitely figure out how to create some time and space for really quickly. What is your iGEM project about? Like, what's the technical area? Oh, Javier, are you still there? Oh no. Okay, hopefully Javi comes back. We'll wait for Javi. Paula, you want to go next?
Okay, thank you. Hi, David. Thank you for all the lectures. I wanted to ask you, like, I registered myself from Bogota, Colombia, but I will be traveling to London, like initially for a month, but I'm not sure how long I will be there. So in case I return, like I was planning to be part of the London node, but in case I return to Colombia before the course ends, can I change like the node to a one in my time zone?
Yeah, I think that should be fine. What I would do is just reach out to both nodes and explain your situation. Again, and I appreciate all of you for your patience and flexibility with us. This is our first time setting up this structure, so I think that should be totally fine, though. You may find that if you start out with the London cohort, that you build relationships with them and want to stay, working with them. If you're really ambitious, you could potentially hop into both meetings to start, and then kind of see which one you're most connected to. So. I'll defer to you a little bit on what feels right, but I think in general, we should be able to accommodate, if you wanted to transition or be a part of both, given your situation, I think that's something that we could work on.
Okay, yeah. Perfect.
Yeah, as Inger says, we will work something out. Yeah.
And my last question is, I want also like to travel to Boston and I'm not sure if there is like any open campus tour to MIT or to MIT Media Lab that like I can
it's a good question. For open campus tours, I'm not sure. I think you could talk to the admissions office maybe to see, but If you just continue building relationships with, our TAs and our team here, I think there's a lot of how to grow students that visit the classroom throughout the year. Global listeners, if they're in town, they'll send a message and be like, "Hey, I'm in person. Can I come to the lecture?" And almost always yes. So, definitely come. If you are in person, again, we'll see how the next few weeks play out. This is we're gonna wait and see. Like we've never had this much demand before for the course, so we have to be. We'll kind of see over the next few weeks what happens. Again, there's always a lot of excitement on day one, and then it kind of shrinks down, over time. So, but in general, though, I think, if you were to come visit like later in the semester, as an example, and, you continue to do the homework and you're engaged with our TAs and our node leadership and so on, it'll feel pretty natural. And you could come visit us in person. And then if you've got new friends that are instructors or students that are here, I'm sure there'll be people that'll be very happy to meet with you and take you and show you things. So
Thank you very much.
great. Thank you, Paula. Okay. Ahmad.
Yeah, hi. Thanks for
Hello.
amazing work you are doing. Thanks very much. Okay,
Thank you for joining.
My question is, I'm interested in, especially in protein designing field. So, what level do you think we are going to reach in this course? So, just introduction to the programming, or we actually will design proteins and
Yeah.
we'll be able to test it in the lab, like binding affinities or expression rate.
Yeah, great question. Great question. So, our hope, again, we've got 2 weeks that are dedicated to protein design. The first week is going to be about pipelines and showing you a lot of different types of tools. And so you'll have an opportunity to kind of, familiarize with some tools and do some homework in week 1. Week 2, we're going to guide you towards the group project. So specifically around the lysis proteins, because that's a big project that we've been all working on together. But I think for you, again, it sounds like you've got some expertise and some particular interest. I would encourage you to think about this as a final project, right? And because we have very, very deep technical expertise in our faculty and also in our TAs. So I think, if you're interested in this area, I would encourage you to go deep. And, we have TAs that are literally, the ones at the front of the field building these tools. So they can, go deep with you. Okay, so I would encourage you, over the course of the 2 homeworks, you might get to a certain level of depth, but if you've got passion in this area, do it for your final project. And I would start this week. Start thinking about it as it relates to a governance framework for areas you want to explore. And then once we start thinking about final projects, go in deep. And there's definitely TAs I think that would be excited to support you in that journey.
Yeah, thank you.
Great. Assem.
Yeah, can you hear me?
I can hear you, yes.
Yeah. So it's Asim.
Asim.
Yeah. First of all, thank you, Kong, for the opportunity, and I really like the energy that you're having. So my question is it possible if I do my wet lab in my own city where synthetic biology is already happening, and there are some people that were iGEMers as a participants and as judges as well. Maybe you might know Sarah Khan from Peshawar, Pakistan. So if I'm asking if I can do my wet lab in my own city in that particular laboratory as a node.
Yeah. So basically, this is it's very exciting for me to hear all this because again, I think there's it shows I think we definitely are very excited to grow this network and want to set up more nodes. So I think part of what I would propose for you and anybody else that wants to set up a node for this year, we can support those 8 official nodes, as I mentioned, because there's an experienced TA that is there, that's on site, that knows what they're doing, that's been a part of How to Grow in many cases for many years, or has a lot of deep technical expertise. And so we really, really want any experimental work to be done in connection with a TA that is experienced in the class. So again, I think you're in Pakistan, so potentially, it's geographically not perfect. I wish we had a node a little bit closer to you, but it will likely be, either Korea or Japan might be the closest to you. So what I would do is, and actually, just an example, we've got Patrick here who's running the Korean node. the South Korean node. Talk with whoever is running the node, and you potentially could do some of those experiments. But again, I want to be as mindful and as careful as possible because like the most ideal situation would be for you to take the course, okay, kind of go through the whole process, and then you can establish a node after you've actually like gone through the whole thing yourself, and then you can be a TA that is leading a node next year. Okay, that would be like the most ideal situation. Now, that said, there may be experiments that you can all of the experiments that are part of the course, they're all biosafety level 1. There's nothing that we're doing that's particularly hazardous or challenging. But again, we would not officially, quote unquote, be supporting you in experimenting locally, right? So I think if you were to do some of those experiments, I would want that to be, with awareness to whatever node you're attached to. So again, just let's say, for example, if you were working with Patrick in South Korea, he could offer some, advice or some guidance to you if you were going to be doing it locally. But that would be, just you kind of doing those experiments, not officially like kind of as a part of the course in some ways. It's sort of a gray area that like from an MIT perspective, we want to really kind of be as, connected as much as possible to the sites that we've got experienced TAs. But again, so I would let you, speak with the whichever node you work with and whoever's leading that node. And Patrick might be actually a really good person. And then you can kind of chat and figure it out a little bit there.
Got it. Thank you so much.
Yeah, but again, it would be great for you to stick with the whole class and then help lead a node next year. That would be the most ideal that we would love to do with you.
Cool. Thank you.
Great. Thank you so much. I think I have answered all the questions. Is that true? Any other questions?
Javier rejoined, so if you had a question for him.
Oh, Javier, are you back? Can you hear me? Well, still having problems.
Yes, sorry, my internet connection. I don't know if you can hear me.
Yeah, we can hear you, Javier. So I'm not sure if I answered my question.
Don't worry.
Did I answer your question?
Yes, actually, yes. I will contact to Sergio so in order to coordinate with agent, especially in Latin America. Thank you, David.
Okay, great. Thank you so much. Okay. All right. Are there any other questions before we start to wrap up here?
David.
Yes. Who is this?
Vancouver, yeah. Just wondering for tomorrow's recitation, do we join the same Zoom link or?
Yes, same Zoom link. It'll be the same Zoom link, and try your best to join if you can. If you can't, so I should say too, at 5 o'clock Eastern time, when the MIT students are going to be, in person doing a safety training, the global TAs will be hosting an office hours for 30 minutes. So we'll actually have another time for more question and answer at 5 o'clock Eastern time tomorrow.
Thank you for the conversation.
All in the same. Everything for the class, for the main course, is all going to be in that same Zoom link. The recitations, the lectures, all in that Zoom link. For the global recitations that happen, those will be separate Zoom links that will be organized by those local nodes.
Awesome. Good to confirm. Thank you for holding the session.
Great. Thank you so much. Okay, any other questions? All right, I'm going to stop the recording then.